I'm happy that I can invite all of you to one very exciting journey around the world, and on this journey we'll meet several of our valuable friends, phytoplasmas. And in order that we'll be able to hear all presentation and still have some time for discussion, I really ask all the presenters to keep up with their time. And now, from here, from Oman, from Muscat, we are moving over the Arabic Sea to India. And our first representation will be from this part of the world, and first two will be presented by Govind Prestaparao, and I would like to invite him to first tell us something about evidence of Phytoplasma association with Lupha cylindrica phyloide from India. Please.
Very good afternoon. Thank you, Arina and Nicola. And I would like to thank the organizer to give me this opportunity to present my findings for records of new phytoplasma strains in India. However, it's not a big, very small, simple record of new phytoplasma from cucurbit and an ornamental. But as again, it's very important if we concern epidemiology. As you know, India is again have a wide diversity of plants and phytoplasma strains. And for the last, in last 2 decades, we have identified more than 100 phytoplasma.
Please put in
yes,
presentation mode.
In last 1 decade, we have reported more than 100 phytoplasma from different plants, belonging to 12 phytoplasma groups in India. And before going on the final results of this presentation, which is a new record of phytoplasma belonging to 16 SR second D group on a cucurbit. It is a very common vegetable crops in India, Lupha cylindrica. I would like to give a brief for the audience, the importance of phytoplasma in India. You can see we have a wide distribution of phytoplasma in different states of India. And so far, we have recorded presence of either one or more than one phytoplasma strains in more than 23 states of India. And nearly 200 plant species are listed as phytoplasma host, 55 belonging to 55 angiosperm families and 2 gymnosperm family, belonging to 12 phytoplasma group. But really we are suffering very severe losses in horticulture crops, especially vegetables and seasonal ornamentals, except then sesame, phyllodes, sugarcane, grassy shoot, and some of the very important vegetable crops like brinjal, tomato, and most of the cucurbits. Some of the stone fruit crops, most important is peach in India. And ornamental is really is the major concern phytoplasma, because India we have a seasonal wide diversity of seasonal ornamental species we are being grown for ornamental purposes in our institute premises, hotel premises, on the roadsides. And this is the major source of phytoplasma host, seasonal one. Because of very, and several insect vector we have been reported. So this is again very, very serious. And for the last 2 years, we have been surveying bamboo. Bamboo is again becoming a very important economic crops in India, and especially northeast part of India. And it is a very serious concern. We have reported 3-4 phytoplasma groups. So bamboo is also becoming an important concern. And out of these, as all over the world, Aster yellows is the most widely distributed group, followed by second our D group at both CFP aurentifolia and Candidatus Phytoplasma stellaceae strains are available, and followed by trifoli, that is in our 6th and 11th group. As I told earlier, we have already identified more than a dozen of vector species, mostly belong to leafhopper, and some of plant hopper, at least 2 plant hopper species. We are very potential vectors of most of the phytoplasma strains in India. I have listed the names. I'm not going in detail. Just for a brief information, I'm presenting this information. And very recently, we identified a new leafhopper vector, Muquaria splendida, which is inhabiting bamboo plantation and very efficient vector for transmitting bamboo strains, phytoplasma strains associated bamboo species in India. Here you can see the real diversity of phytoplasma. You won't find a single crop which is not being infected with phytoplasma in India. You're starting from fruits, ornamentals, medicinal plants, vegetables, cereals, spices, pulses, oil crops, cash crop, palm, wheat, and trees. So this, and this is really very, very emerging. And every year we have been reporting new host species, new vector, and that is a real, now is a big challenge for us to, and there is no resistant, no pure, in any of the crop, we don't have pure resistant, this genotype. So that is again a very important, how to manage these phytoplasma diseases. Now come to the vegetable crops, We have again India, we grow all sort of vegetables now. And so far, we have reported 22 vegetable crops as a host of phytoplasma of 7 groups in India. And out of these, little leaf disease of brinjal is the oldest one and is still causing severe losses in most of the different states of India, and mostly dominated by 6D subgroup, CFP trifoli. Followed, and for the last, we reported first time 2018 tomato big bud disease in southern part of India. But within 2 years of time, it is distributed everywhere. Now, all states, because tomato is again a very, very important vegetable crop in India. And we recorded incidence of tomato big bud disease, and we found association of second D group with tomato big bud disease in all states where tomato is being grown. And as you know, tomato is again a host, very, very popular host for different types of virus diseases. So most of the time, we got mixed infection of phytoplasma strain with Beguma virus. So again, when there is a mixed infection, there are chances 100% losses are there. So again, is a very, very important concern. And then followed by cowpea, cauliflower, the chili. Chili is again very important, where you got mixed infection. Generally, we find mixed infection of phytoplasma with different types of viruses, potyvirus, cucumber mosaic virus, and different types of Beguma viruses. And then cucurbit, that is today I am going to discuss a new phytoplasma strain we identified from a cucumber in India. Earlier, we have also reported, at least you can see, 4 different groups from bottle gourd, beetle gourd, cucurbit, and pumpkins. Here you see, just for information, I have listed, put the symptoms of different important phytoplasma disease on vegetable crops. As I told you earlier, so far we have recorded phytoplasma diseases in 22 vegetable species belonging to 7 groups. But major groups we identified, they are from 1st, 2nd, 6th, 11th, and 14th group. And out of these, I told brinjal is the most, brinjal little leaf, tomato big bud, cowpea, proliferation, and cauliflower is the most important, followed by cucumber. Especially come to cucumber, you can see already we have listed, already reported 6 different cucumber species as host of different phytoplasma strains from India. And in the present study, we have identified a phytoplasma strain from Lufa Selendica. Earlier there was a record, but the author didn't characterize only on the basis of symptom and primers. He confirmed the presence of phytoplasma, but he couldn't confirm the group and subgroup. So in the present study, we surveyed Uttar Pradesh is one of the biggest province in India, and we surveyed Ayodhya is a major area in central UP, where we surveyed campus of a university and the university campus, where all different experimental fields are there, and we found 5 to 12% incidence of phytoplasma suspected symptom on Lufa Selendica. That is again a very common vegetable in different states of India, including Uttar Pradesh. We have 2 different common species of Lufa in India. You can see one with very smooth skin, that is smooth Lufa gourd, and one is with ridge gourd. Both are very, very common, but we recorded particular phytoplasma suspected symptom in sponge gourd, that is smooth Lufa Selendica, very commonly known as Turei in India. And we attempted identification of phytoplasma. We extracted DNA. Please see the symptom. We recorded, this phyllodes and flat stem, and which is broom symptom in most of the grown plants of Lufa Selendica in the fields of the university. And we attempted identification by extracting DNA, and we attempted the universal primer, P1, P7, and R16 primer, and we got very good amplification with both set of primers, and we amplified the R16 product of 1.2 kb and submitted the sequence in GenBank. And when we compared, we found that it very closely matches with the strains of 16SR second group. We also attempted phylogeny. This again confirmed that our particular Lufa strain belongs to 16SR, matches clustered with earlier reported 16SR second D group. Again, we confirmed with virtual classifier, and again this confirmed that our particular strain belongs to 16SR D group. That confirmed that the Lufa isolate in the phyllodes and which is broom isolate in the present study belong to second SR D group. And I told this particular group, already we have been reported in earlier number of other vegetables, ornamentals, and other seasonal crops. So it is very, very widely prevalent strain after Asterellus group in India, in all parts of India. So again, it is challenge, and several leafhopper vectors have been already reported. In the same university campus, in earlier years, we have recorded the same strain on other ornamentals and other crops, and that clearly indicates that must be potential vectors available in those fields which are capable of transmitting these particular strain from other crops to this particular crop. And we again, I have not included in this particular study, after we have identified the vector, and that's again Impesca species, which is again, we identified the same phytoplasma strain. But still, our study, we would like to confirm whether it is a confirmed vector, natural vector of this particular phytoplasma. So total, as a conclusion, I can conclude that phytoplasma diseases are very, very emerging in different types of vegetables in India, including cucurbit. And in this particular space, earlier there were 2 reports available of second D group, not second D, second group from Taiwan and Brazil on the same species of loofah. But India, it is only, simply confirmation of phytoplasma presence. They, earlier author, they could identify the strain. But here we confirm that the particular strain is belong to second D subgroup. And that is a new report from India. And as I told, it is a very wide prevalent strain. Many host species are there. We have also recorded several weed. Weed are again playing a very important role as a natural reservoir for natural propagation of phytoplasma. As you know, India in all around the agriculture field, lot of weeds are growing naturally, and they are very, very, this natural host for different phytoplasmas. And vectors are also there. So it is quite, it may be quite possible that these bacterial species, they can, vice versa, they can transmit the phytoplasma from weed to, crop species. And when crops are harvested, again, they survive on those weed species. So this is a common, the potential, because of potential weed reservoir, the life cycle is going on, and that is the natural, we have regular recurrence of phytoplasma from, different crop species in India. So that is all about it, very small, but it is very important in the concern that regularly we have been recording addition of new hosts for especially first and second D group in India, and that is a very, really of very epidemiological significance. We have to look after more and more potential leafhopper species which are real responsible for transmitting these strains in different states of India. And definitely we have to take every care for management strategy. And now, since there is no resistance, now we have full genome sequence. My colleague Amit Yadav, yesterday we have full genome sequence available for our group. We have also sequenced second D group from sesame in India. And again, Amit Yadav, he sequenced a full genome sequence from Parthenium, a weed species. So luckily now we have a full genome sequence available. So we have more and more information about the genome of these phytoplasma. So in future we can utilize these information of genes to further with help of transcriptomics study and other molecular studies. We have to identify the genes which are really responsible for now we are also working on what is the particular effector proteins for this particular phytoplasma strains, and this will give a lot of our plan for future studies, how we could manage these important strains in India. So I would like to thank our colleague from Oman, Dr. Abdullah Al-Sadi and Professor Ali Al-Soubi for their kind support in waiving our station and my university. Earlier I was working with central government at IRI New Delhi. So I established a very good lab and prepared a very good team, young team. They have started working on phytoplasma identification in different parts of India. But last year I retired from service, and again I joined a state university. And again I'm starting to develop a good facility for students at that particular Uttar Pradesh. That is again a big province, agriculture province, where we grow all diversity of crops, cereals, vegetables, sugarcane, oil crops, seed, so all crops are there. So again, let's see, I think in next 2-3 years, I would able to develop again a very good facility at Gorakhpur for having more and more information from that particular part of India. And again, I thank all the co-authors who are involved for survey work, for lab work, and for writing this manuscript. Thank you so much.
Thank you, Govin.
And just
We have no time for question. You use more than your time. Please go in again with the next presentation.
yes, I'm
Okay?
going to yeah.
Please.
Here's my again.
Go ahead,
Okay.
please.
Before going to the next one, just again a simple, we are publishing now. You all must be having a phytopathogen molecules journal. We are being published for the last 15 years, and I need your support. No, it is already a Scopus index, and we are trying our best to have ISI indexing at earliest possible. But no, there is Springer and other. We are also approaching. There are no other important recognized publishers to take up our journal. So I think in next 2-3 years it will be being taken by Springer or other publishers. So I just request please send your publication to make our journal more effective, and more popular. Thank you very
Good.
much.
Thank you. Go.
In same continuation, I have another presentation of one of my PhD students. They couldn't come because of some health issue. So I'm going to present on her behalf. Again, there is a report of phytoplasma strain from new ornamental species in India. Yes. In earlier presentation, I already discussed that ornamental is again a very common, popular host for phytoplasma strains in India, and we have recorded at least 50 ornamental species of different phytoplasma groups in India. So, Surabhi Mitra is one of my PhD students, and she worked on identification of phytoplasma. And she identified phytoplasma strain causing little leaf and witch's broom of Tecoma stance. It's a very popular here. I also found this plant in Oman campus, university campus, just outside this building. So it is very popular with very, very important on the roadsides you can see. Very popular for its bright yellow flowers, and belonging to Bignoniaceae family, very commonly known as trumpet flower. And this also plant is also very commonly used for different as a herb for use in different medicinal purposes, and very commonly used for landscapes in different parts of the world, including Asia. So again, in the same campus where I discussed. We described a new strain on cucurbit. During the survey, 1 year after, we recorded phytoplasma suspected symptoms on Tecoma stands in the same university and university campus, Ayodhya, Uttar Pradesh, in 21-22, and we isolated DNA and used the same set of primers, universal set of primers P1, P7, and R16 for the identification of confirmation of phytoplasma. And we identified the similar phytoplasma strain which we identified in the cucurbit plant, just discussed in earlier presentation. So again, it is the first record of presence of this and this is a new host of this phytoplasma in India, not in India, all over the world. You can see the symptoms, very, very declining symptoms, witch's broom symptoms, little leaf, and this is the healthy one. So it is very commonly, incidences we recorded not much, up to 5%, but since it's a long, it's a landscape, so at a distance one can see that is on this row, these much of plants are infected. So that way, there is no flower, very few flowers are there, and after within a 1 month or two, it won't survive. So similar, we submitted the sequence, and we did phylogeny, blast analysis, and virtual RLPP, and this all indicates and confirm that phytoplasma stain associated with Tacoma belong to a second, 16 SR second D subgroup, that is CAP Australasia. So again, no, this all, whether it is vegetable or ornamental, if the similar phytoplasma stain is infecting, this is again a very serious epidemiological concern. That because regularly we grow, no, in the same field, we crops as we grow vegetable, then cereals, and then other horticulture crops, other oil crops, and the same area because the insect vectors are common. So it is very, very quite prevalent. Then these potential already, especially Hicimonas, Orosius, Aciniatus, and Amphoesca. In this area, we have reported as a natural host for second D group. So it is very, very common, and it's also several weed species like cannabis. One of my next presentations, they will discuss. Some potential weed species are very, very, again, very important as a natural host reservoir for this particular phytoplasma stain, like Datura, Stramonium, and Cannabis, Parthenium. These are the very common weeds grown in our agriculture fields near around, and very, very common house for these phytoplasma stains. So now we are working on epidemiology. What are the potential weed species? What are the potential leaf hoppers which are involved in natural transmission of this phytoplasma? So again, this is the new finding in India, and again we are working on different epidemiological aspects and other management, how to manage these phytoplasma stains in India. Thank you.
Thank you, Govin. Some question, please? In the room? No? Online, we have questions? No? Okay. Thank you again, Govin.
Thank you. Thank you very much.
Thank you. And we are staying in India, and I would like to invite Smriti Mal to tell us about identification of 16S2 group phytoplasma strain associated with witch's broom of spinach and hemp in India. Please.
Thank you. A very good afternoon to all of you. Before start my presentation, I would like to say thanks to the organizers of this event, especially Professor Osunta Battasini Madam and Ali Sobi sir, and for giving me this opportunity to present my work here. Today my presentation topic is identification of 16S ribosomal second group phytoplasma related strain associated with witch's broom of spinach and hemp in India. Myself, Dr. Smriti Mal, Assistant Professor, Molecular Plant Pathology Laboratory, Department of Botany, Deen Dayal Upadhyay, Gorakhpur University, Gorakhpur. Well, as we all know, that phytoplasmas are characterized by wall-less pleomorphic bacteria, insect vectored, and cause diseases in a wide range of plant species globally. They have the smallest genome and have limited metabolic pathways. And in present scenario, over 1,000 diverse plant diseases have been reported worldwide, including vegetable crops. Here my concern is for vegetables because I'm working in vegetable crops. So vegetables are short duration crops grown during different seasons of the year, and which fetch high economic returns. And as we all know that they are the excellent sources of nutrients, dietary fibers, phytochemicals, and vitamins, as well as contribute towards lowering the risk of heart diseases and stroke, and also helpful to boost up immunity. But in last few decades, several abiotic and biotic stresses attack vegetables, and the phytoplasma associated diseases represent one of their major constraints in several parts of the world, causing significant losses in production, yield, and quality. Phytoplasmas diseases of vegetables mainly affect plant species belonging to the botanical families, especially Apiaceae, Asteraceae, Cucurbitaceae, Fabaceae, and Solanaceae. Now, this is the geographical distribution of phytoplasmas in vegetable crops. Here, we can see here that around 47 countries have been reported that are associated with phytoplasma infections on different vegetable crops. The phytoplasma in the aster yellows group, that is 16SR first, are predominant across different genera, followed by the peanut witch's broom, clover proliferation, and stolber groups. Interestingly, among the 4 subgroups of aster yellows, that is 16SR first A, B, C, and X, infecting vegetable crops, the 16SR first B subgroup is the most predominant worldwide, whereas the subgroup 16SR first C seems to be restricted to the Asian countries, maybe because of the presence of potential vectors in other part of the world. Now, phytoplasma virus in mixed infection in vegetable crops. Here, I have quoted here to just for information that some mixed infections are also reported on vegetable crops, and some common examples are like tomato crop, several reports of virus and phytoplasmas co-infection on vegetable tomato crop, that is association of 16SR's third group with 2 bigamovirus and tomato leaf curl virus reported from Mexico, and other mixed infection report on this brinjal crop also belonging to brinjal crops, that phytoplasma belonging to 6th group and along with bigamovirus. And some reports are also from Karnataka, India, about the mixed infection on vegetable crops. So the presence of phytoplasma in mixed infection with viruses, this is an emerging threat to vegetable production worldwide. Now, in this scenario, the next important leafy vegetable is Spinacia oleracea, commonly known as spinach, belongs to family Chenopodiaceae. Habitat native to Southwest Asia, widely distributed and cultivated throughout the world. Parts mainly used are leaves, and nutritional and medicinal values like helpful in lowering body weight, boosting immunity. Spinach is an excellent source of lutein and an antioxidant known to protect against age-related eye diseases. It is an excellent source of folate and different types of vitamins, good source of iron, magnesium, and riboflavin and potassium. Now, experimentals, samples of spinach and hemp, that is cannabis sativa, that showed symptoms of witch's broom were sampled from Ayodhya region of Uttar Pradesh, India, during October-November 2021 to 2022. And the collected samples were analyzed through PCR analysis, and then sequence analysis were performed, phylogenetic study were attempted, and virtual RFLP were attempted to characterize the phytoplasma. This is the symptom that we were observed during collection of the sample, spinach witch's broom, and in the same field, cannabis witch's broom were also observed, and both samples were collected for further analysis. This is the PCR result. We got the positive result of phytoplasma. Phylogenetic tree from this, both isolates, spinach witch's broom and cannabis witch's broom, shared highest similarity with 16SR second group phytoplasma. RFLP were performed using 17 restricted enzymes profiling, and here we also found association of second group, subgroup D phytoplasma. Now, results of witch's broom symptoms was recorded on a spinach plant with 2% disease incidence. The association of phytoplasma was confirmed by PCR amplification, and then 16S ribosomal RNA sequence of Spinus witches broom and Cannabis witches broom revealed 100% identity with those of Candidatus phytoplasma australisia strains. Phylogenetic relationship again confirmed this association, and then both Spinus and witches broom isolate shared identical restriction profiles to the reference phytoplasma strain of 16SR2 T subgroup phytoplasma. On the basis of this study, it was concluded, it was confirmed the association of 16SR2D group with Spinus witches broom and Cannabis witches broom disease in India, and this is the first report of association of hemp, that is Cannabis sativa, as alternate host of 16SR2D phytoplasma on spinach. Previously, spinach witches broom has been reported from here in Oman by Al-Subi et al. 2019, but the association of alternate host is the first time first time reported from India. In the present study, Cannabis sativa has been suggested as natural putative propagation host of 16SR2D subgroup of phytoplasma. And some other reports are also available that shows that Cannabis sativa is one of the major potential reservoir of phytoplasma, not only in India, but in other parts of the world also. So we have to look at, to eradicate such weeds from field and to use some integrated management practices to eradicate such weeds. Thank you. This is the banyan tree, which is present in my university campus. This is 150 years old. Thank you so much for giving me your precious time and listening to me. Thank you.
Thank you for your presentation, and I'm asking the audience for questions, or if there is someone online. No questions? Okay, then thank you again.
Thank you.
And let's make a jump over the Strait of Hormuz.
Yes, I invited now to present a poster. I remember that you have 5 minutes. Sayed Alireza Esmail Saed Hosseini, sorry for the pronunciation. Occurrence of a Candidatus phytoplasma erantifolia strain associated with the Euonymus japonicus fasciation. Please say it.
Okay. Good afternoon. Euonymus japonicus phytoplasma reported from Iran for the first time as a peanut witches broom group. Many of the symptoms of fasciation or flat limb were observed and reported from Iran as Cartamus tincturus, as a peanut witches broom, and also 16SR9 subgroup A and Cartamus. Stem fasciation symptoms were observed up to 8.4% in Euonymus japonicus grown in park and gardens in Yazd area, especially grown Euonymus japonicus in parks and gardens. And the result of molecular study with universal primers in direct and nested PCR showed that Euonymus japonicus plant samples infected with phytoplasma belonging to peanut witches broom group in group 2. This is the symptoms of fasciation or flat limb in Euonymus japonicus, and also phylogenetic tree showing the Euonymus japonicus fasciation in Yazd belong to a peanut witches broom group. This is the first report of phytoplasma associated with flat limb of Euonymus japonicus in Iran. As you know, peanut witches broom is widespread and distributed in all of the country and cause damage disease to different species of plant, fruit, weeds, and others. As Euonymus japonicus grown on a margin of parks and gardens and may have an important roles in the epidemiology of and spread of Candidatus phytoplasma australasiae phytoplasma as reservoirs. Thank you very much for it.
Thank you, Sayed, for staying time and for your presentation, of course. And I don't know if there are some questions. Assunta, please.
Thank you, Alireza, for your presentation. I take the occasion to just suggest to the other speaker to stick to the terminology as Alireza was using and some other. We all know well that phytoplasma are associated with diseases. So please avoid to say and to write causing diseases. We didn't fulfill Koch postulate yet. So we must be careful. If one day somebody found something else, we have our paper wrong. If we write associated, our paper are correct. So this is just not a question for you, but just thank you because you use the appropriate wording, and also from now possibly try always to add the Candidatus phytoplasma species to your ribosomal group in order to be able to discuss with the others. Thank you.
Thank you, Assunta. I have another question. Online, Fabio? We have something? No. Okay. I have a question. Do you know if associated to the fasciation may be some viruses in this plant, or do you not have idea?
We have reports of flat limb or fasciation due to plant viruses, and also many researchers think about insects.
Okay.
But in this case, we cannot observe any insects, and also we don't check any viruses.
Okay. Okay, thank you so much. Thank you.
For the next presentation, we need to cross Saudi Arabia and go to Jordan, and I'm inviting Sultan Qaboos Abu Alush to tell us about phytoplasma-associated diseases in stone fruits, pomegranate, and grapevine in Jordan.
Okay, Assalamu alaikum. Good afternoon. I would like to express my special thanks for the organizer, Dr. Assunta Berticini, and her teams for their efforts and invite us to this great effort, and also Marina and Fiore to introduce me. This is okay? Okay. I have to repeat, or it's okay? Okay. And it's a pleasure to be here right now to share the findings of PhD project about the phytoplasma associated disease in stone fruits in Jordan, in stone fruits, pomegranate, and grapevines in Jordan. And Nida yesterday summarized the current situation in Jordan, but we have also to state the major problem in this issue, although the importance of the phytoplasma associated disease in Jordan and the significant impact. There is still information, very big information gap in this issue, and lack information about their epidemiology and the factors and all related aspects. So previous study were really too short or too few. Based on that, the current this study was initiated aiming at survey phytoplasma associated disease of grapevines, almond, and pomegranates in Jordan, and to identify also the 3 crops were selected based on their economic importance and the observed symptoms in the previous season, and to identify the phytoplasma associated with such diseases, their putative vectors, and reservoir plants. Before going on methodology, I want to highlight some facts about the almond and the other crops. Almond is a minor crop in Jordan, exceptional because the stone fruits is major crops, but this is gaining a huge popularity right now, and you can imagine that the 1 kilogram of a fruity green of almond costing for 12 or 15 for pre-harvesting for 15 Jordan dinar, more than 20 euros. And the larger consumers, pregnant women. I don't know why. And with the grapevines, very promising sectors. And we exporting a lot. With pomegranate is one of the oldest fruit trees in Jordan and mentioned in the Holy Quran. And we have inherited orchards, and by law this inherited orchards became the natural reserves in Jordan. Back to the methodology, and with almonds in around more than 7,000 plants in 23 almonds orchards in northern Jordan were surveyed from June to October in 2020 and in January 21. And around 140 symptomatic almond trees and 16 symptomatic plants of weed species were samples, and we collected around 126 Ocorinca species. With the grapevines also in the same period, from June to October, more than 22,000 plants in 11 table grapes and 2 wine grape farms. We don't have a lot of wine grapes just because we are an Islamic country. And around 152 symptomatic grapevines and 22 symptomatic bindweeds also were collected, and all in addition to 1,173 Ocorinca adults were collected. With pomegranates, the survey lasted from June to November, and more than 5,000 plants and 22 pomegranate orchards were surveyed, and we collected, or we sampled 112 symptomatic planted trees and 30 symptomatic plants of 11 weed species. For example, the other procedures are well known, so I will cross quickly with this, but I have species attribution were compared with the candidates phytoplasma species references strains, and we use the IFI classifier to attribute the group and subgroup. What we have with almonds, with almonds based symptomatology, we categorize 5 groups, and each groups were associated with certain 1 or 2 candidates phytoplasma species that will be shown later. The first group was witch's broom, yellowing and dieback, and then we have witch's broom, yellowing and leaf rolling, slim leaves, flat stem, and also we have very interesting symptoms in terms of early flowering and evergreen. This is observed in early of January in snowing area. The pre-harvesting or the early flowering season starting in March or late of February, but in this case, we observed these symptoms in the early of January. It was also, as you know, the stone fruits, almond, and others also starting flowering before leaves. But we have both in the same time in very cold season. So what we have? So 7 candidates phytoplasma species were detected and identified belonging to 6 phytoplasma groups and with different subgroups and variant of subgroups. For example, we have candidates phytoplasma astres were associated with early flowering and evergreen. And also we have ulmi with the same symptoms in the same area. We have with group 5 subgroup A and variant of subgroup A. Then we have candidatus phytoplasma omanense in 2 areas associated with 2 kind of symptoms, slim leaves and witch's broom, yellowing and dieback. And here we added a new subgroup B, based on the coefficient. Interestingly, also we have, even with low infection rate, we have Candidatus phytoplasma faunicium, but this even with even we're talking about the low infection rate with this destructive diseases, but we have to consider that with available efficient vectors, this will be very spread quickly and very destructive one. And also we have Candidatus phytoplasma solani associated with this, which is prone dieback and yellowing. And we have Solani were detected and identified in Hofa. Hofa is one of the major cultivation area of almonds. In the same area, we have Aurantifolia. Then we have, very interesting, we have Candidatus phytoplasma beri. This is never reported in this crop. Beri reported in the stone fruits in peach in Italy, but not with almonds. Another important issue that we have to highlight or consider, because we have Astris and Olmi in Ajloon or in the north. This is agroforestry area surrounding by the forestry area. As all of you know that this species could be mortality factor for the woody plants. And also we detected Omanense with one weed species, Amarantha species. Regarding to the insects, we collected around 12 insect taxa belonging to six insect families, and the infection rate ranging from 16.7 to up to 100%. Only we detected 2 Candidatus phytoplasma species with the insect, Candidatus phytoplasma beri, and we have Candidatus phytoplasma astris. We have a subgroup variant of subgroup R in this. Lodelfax, Cicadolina bipunctata, and Cacopsella bidens were infected with this, and the other species were carried the astris, Candidatus phytoplasma astris with variant of subgroup R. With the grapevines, also we have the typical symptoms: leaf rolling, yellowing, and reddening. And in the first one, we have the leaf rolling with table grapes, and the other with wine grapes. And this is everywhere. It's not restricted for the certain cultivars, table or wine, but this is everywhere. So, also with table grapes, we have 4 candidates. We detected or identified 4 Candidatus phytoplasma species: Omanense, Solani, and Aurantifolia. And with the wine grapes, we have also Aurantifolia, Astris, and Solani. With the insects or the collected fauna, with the reservoir plants, we identified Candidatus phytoplasma Aurantifolia with the bindweeds that collected from the table grapes, and Solani, only Solani from the bindweeds that collected from the wine grapes. Regarding to the insect species or the collected fauna collected, we have almost 9 insect taxa with wine, including Emboasca, Psamotetex, and Orosius cellulosius. This is first reported in the country. And also we have Echilidus mundus and others. And also we have 6 insect taxa we collected insect taxa from the table grape orchards. Okay, attribution to species and taxonomic groups. Interestingly, we have more Candidatus phytoplasma detected and identified with the insect factors than that detected in the plants. Here we have more beri and we have Omanense, and that is maybe we have good explanation for that. And with the table grapes also we have Astris, Solani, and Solani. With the pomegranate, we also reported several kinds of symptoms or phytoplasma-like symptoms, including little leaf discoloration, yellowing, reddening, and witch's broom, which the majority of the symptoms were observed in everywhere, exceptional the witch's broom were associated with one cultivars in certain area. This is very traditional cultivars. Okay, what we have, we also with pomegranate in the first traditional area, we detected 4 candidatus phytoplasma species, including Astris, Orantifolia, Sulani, and Ulmi. And in the other area, we have Sulani and Ulmi and Astris, and also in the desert, we have Sulani, with all the collected weeds or bindweeds were infected by candidates phytoplasma Orantifolia, variant of subgroup C. In the total or overall, we can conclude that the candidates phytoplasma Sulani is the domain of candidates phytoplasma species by 52%, and followed by Orantifolia by 21%, and candidates phytoplasma Ulmi by 60%, and the final one is candidates phytoplasma Astris by 10%. Also, we collected more than around 11 insect taxa, and 4 of them firstly reported in Jordan. Nida yesterday mentioned that we have 140 species listed and identified by a PhD student, but just listed as their work for classification. And this first work, this is the first work conducted or detected phytoplasma with insects. So we have in our study, we added 8 insect species to our Ocorynca fauna. So right now we have 122 insect species of Ocorynca, in addition to the Psyllidae. Okay, only we have Astris and Sulani were detected with Macrostellus and Zygidinia zohrab and Psammothetex striatus, and Balicluta incieza were carried the candidatus phytoplasma Sulani. Before I conclude my study, I want to highlight some information or the facts that obtained from our studies. Almond disease complex associated with multiple phytoplasma, including candidates phytoplasma piri, omanense, and olmi, never reported before in these crops. Grape yellows phytoplasma diversity and ecology in Jordan are more complex than the previously known, leading to potential risk of disease outbreaks. New pomegranate disease associated with multiple phytoplasma, including candidates phytoplasma Orantifolia and Ulmi, also never reported in this host plants. And we concluded that further studies are essential and must or should be include upscaling survey of fruited trees disease complex in the whole country in terms of diffusion and epidemiology. Also, we have to conduct transmission trials to identify the capabilities or to categorize which vectors and which is just putative. And also, we have their capabilities to transmit the identified species or phytoplasma species. And also, insect population dynamics and diversity through the whole season must be conducted. At the end, I would like to express my special thanks to my lovely teachers, Fabio Qualino, Rosemary Tedeschi, Nicola Mauri, Marta Martini, Piero, Roberto Alma, Enrico, and Jordanian ones. Thank you very much for your attention.
Thank you very much for this really important new informations. You found some usual suspects on both sides, on the host side and on the phytoplasma side, and several new combinations. So a lot of work for next period. So do you have any questions? Yes, please, Sultan.
A very nice talk. Finally, it was a little bit clear, all the huge work you are doing. But do you know, I was impressed. In 2 years, you found more phytoplasma than us in Italy in 30 years. Do you have some explanation for that?
Microphone.
Yes, maybe several reasons for that. Maybe the accumulated experience with my teachers expressed including. They are. This is the goal. Also expressed in this study. Also, we have big team and we have good facilities to survey. We have facilities to survey different regions, different crops, and we have hard workers. And also we worked for 2 years, full 2 years. I don't know, but also we more focus on the survey. But maybe in Italy, maybe focus on certain associated diseases. Or maybe we are too lucky.
Another.
I hope you mean unlucky, not lucky, because a lot of diseases. No, my explanation is I don't know a lot about Jordan, but maybe you have material imported from many parts of the world. You mentioned all the traditional, so you accumulated internal biodiversity and biodiversity from other places.
Yes.
Is it possible?
Yes, Jordan is open area. We are in the central of the open area. There is no very restriction rules for the propagation or seedling materials. And also in the last 2 decades, we imported a lot of stone fruits and also different or many wine grape cultivars. You know, also in the irrigated area, we have big flora diversity. You know, maybe this, and also in the traditional areas, the use of pesticide is extremely low.
You are the best example for the roundtable on quarantine.
Thank you.
But Assunta is lucky too because he have a huge teamwork, I think. No? A lot of people working with you for this kind of work realized in 2 years. Or only? You are only you in the work? In the lab? I think no.
No, we have a good teamwork in Jordan, like they are accompany us during the field visits. And also in Milano, Statali, at Disa, also we have good teams supervised by Dr. Fabio. And at Torino also we have good teams.
Okay. I have a question. Oh, sorry. Sorry. Microphone, please.
I just will add something to what he said. I think also the possibility of finding so many phytoplasmas, because the way how we plant in Jordan, it's like mixed agriculture. Like you find pomegranate with grapes and with almonds, so the possibility is higher. And also based on the survey that was carried out in Jordan by entomologists, they found plenty of leaf hoppers. So this will increase the possibility of finding more phytoplasma.
Thank you. Okay. Did you see other symptoms associated to the presence of Candida phytoplasma piri in almond, other than flat stem?
Yes, there is a size variations, and the infected plants were very dwarf, and they sometimes the different kinds of leaves, and there is size variation with this one.
Okay.
In China, they reported the flat stem with ornamental species or ornamental plant with different candidates phytoplasma. Concerning your questions, my answer is yes. But also you have to know, we also detected and identified peer decline with peer. And I told you, maybe during the lunch, that is with high infection rate with Cacopsella pedens, ranging from 40 to 85.
Yeah, you are doing the transmission trials, I suppose.
Come again, I cannot.
You are carry on transmission trials, I suppose. Or you think to do this?
Yeah. Right now, we have some fun to do further steps, and the transmission trials will be obligatory.
Ah, okay.
We plan to do that.
Okay. Now. Thank you, please.
Thank you.
And
we are moving very fast, both in time and space. So we are already on the Mediterranean coast. And
Thank you, Marina. Now I invited Nicoletta Contaldo. The speak will be investigation of phytoplasma diseases of sweet pepper in the Bekaa Valley of Lebanon. Please, Nicoletta.
Yeah, thank you. Good afternoon. Today I'm going to present the results on behalf of my collaborators, my colleagues. First of all, Dr. Elia Choueri from Lebanon, from the Lari Institute in Lebanon, and my colleague Rayed Aboukuba from the Institute of Sustainable Brain Protection in Bari. And so this is a work carried out in the last years between the two institutes. And first of all, just a brief overview on the current status of phytoplasma disease in Lebanon that actually were carried out in the last 20 years by the group of Lebanese and Lia Choueri and the group of French people led by Xavier Fossac. So the disease in Lebanon dates 2 decades, where almond witch's broom was reported as the first phytoplasma diseases on almond trees. And accordingly, phytoplasma diseases are considered to be invasive diseases of economic importance that affect fruit trees and some field crops in several regions of Lebanon. And to date, the main phytoplasmas in Lebanon are almond witch's broom phytoplasmas, identified on almond and peach trees, grapevine yellows bois noir phytoplasmas, and Candidatus phytoplasma owanense on wine grapes per decline, Candidatus phytoplasma trifoli on tomatoes and peppers, in addition to other phytoplasma infection reported on cactus, belonging mainly to the subgroup 16SR2. Pepper is one of the main vegetables cultivated in Lebanon, and is cultivated mainly in the Bekaa Valley and in the coastal areas of the country, and covers an agricultural area of about 800 hectares for domestic and foreign markets. Previous studies conducted on tomato and pepper cultivation regions in the last decades show the presence of the PTL phytoplasmas, a strain belonging to the Candidatus phytoplasma trifoli, the ribosomal group 16SR6. But in the recent years, the growers see some symptoms in different areas in the Bekaa Valley associated to the presence of phytoplasmas. So they got a severe yield losses. These were the symptoms observed in July 2018, the typical symptoms of leaf yellowing, stunting, and leaf chlorosis. So a survey was carried out on peppers to detect and identify the possible phytoplasma present. Molecular analysis were carried out on 90 samples based on DNA extraction and nested PCR with universal primer F2R2, and some of the samples were positive. In particular, 6 out of the 90 tested samples, so the 6.66% result positive after nested PCR assay. And we confirm by sequencing the presence of 2 different phytoplasmas, Candidatus phytoplasma trifoli, with an identity of 100% with the Turkish strain GRS93, and Candidatus phytoplasma solani with an homology of 99.76% with the Turkish strain Beagle D11. So to further investigate this last phytoplasma, we did also other analysis, because in the previously in the mixed city, this phytoplasma that is the mixed city, that is the city where we detect phytoplasma solani on pepper, is not so far from the Tal Danube city, where it was found on grapevine. So the distance between the two cities is only 6 kilometers. So we want to characterize the strain that we found on pepper. And we did some analysis on other genes. Because in the previous studies, there were other host plants detected in Lebanon, but not pepper. So because you see the different symptoms and that they found in different plants like grapevine, convolvulus, and that they already were reported. So in order to understand the strain that we found on pepper, we characterized the stump gene, and we obtain the sequence identity of 100% with the strain P7, so the ST15 that was already reported in Lebanon in the previous studies. So we confirmed that the strain that we detected in pepper was the same that was reported before. So in conclusion, we can say that this is the first report of Candidatus Phytoplasma solani in pepper in Lebanon. And this finding highlights the necessity to conduct a detailed survey for phytoplasma associated with stunted chlorotic pepper plants in other regions in Lebanon. So, and the analysis on non-ribosomal gene stump further confirmed the presence of Solbus strain in pepper. And even no weed or vector sampling was done in our survey. The presence of Convolvulus albentis have been previously reported. So the presence of Candidatus Phytoplasma solani on grapevine with 1R, only 6 kilometers far from the field where infected pepper were found, and taking into account also that this phytoplasma is detected in insect, is common in the Mediterranean region, this can lead to further analysis on the role of the vector in the transmission between the two insect host species in Lebanon. So I hope that we'll continue to collaborate with our colleagues and in order to explain and to study more on this epidemiology. And again, I'd like to thank all my collaborators and very nice people. Thank you very much for your attention.
Thank you. We are for a couple of questions. Please. Asunta? No. Govind.
The symptom you showed on chilies more, no?
Sorry?
Did you also check for presence of virus? Because symptoms are more looks like virus also.
Yeah. There was in parallel there was the study of the virus, and they got a lot of virus also. Yeah, so they had mixed infection, exactly. That's why only saw a few positive for phytoplasma, because there are also virus. So sometimes it's difficult to detect in the mixed infection. Yeah.
Okay, another question? Online? Fabio, online? No, okay. Okay, thank you, Nicoletta.
Thank you.
Now we will move just a little bit, and I'm inviting Isil Tulum and her talk, Molecular Detection of Candidatus Phytoplasma mali associated with virescence in Narcissus tatseta in Turkey.
Good afternoon, everybody. I'm Isil Tulum from Istanbul University, and today my talk title is Molecular Detection and Characterization of Phytoplasma, Candidatus Phytoplasma mali associated with phyllodes in Narcissus tatseta in Turkey. Just a brief information about Narcissus tatseta, in other words, daffodils. They are geophytes with bulbous structure and grows primarily in the subtropical biome, and one of the oldest known plants in the perfume industry. And you can see it belongs to the Amaryllidaceae family and generally represented with Narcissus genus. If we look at the world distribution, there are some places which is natively growing Narcissus tazetta, and some of the places are introduced into. So mainly it is like Turkey and Italy and maybe Mediterranean part of Italy and North Africa, and other places like introduced as well to Australia, and New Zealand, and some parts in the USA. In Turkey, we have 5 different species, and mainly dominated with Narcissus tazetta and Serotinus. And interestingly, in the winter of 2019, typical phyllody and flower sterility and stunning symptoms were observed in Narcissus tazetta in the south part of Turkey, Mersin Provence. And you may see here the typical phyllody and the stunning in the flowering shape. So these ideas give us, maybe these are the symptoms of phytoplasmas, and we need to check the literature for, is there any phytoplasma reports in the literature. So the first report belongs to Belardi, and they were just deciding it was as an mycoplasma-like organisms, and showing leaf chlorolysis and phyllody and flower degeneration. And 2 recent reports from Iran. One is showing Stolbur and Candidatus phytoplasma solani from the 12A subgroup, and another one is Mahmoudi and his friends, which is showing peanut witch's broom phytoplasma, which is also group 2B. So our aim in the study is to molecularly detect and characterize what phytoplasma disease it is and the symptomatics daffodils. So the material method is like classical, as everybody explained, detect by PCR using P1, P7 primers, followed with a nested PCR with F2R2. And then we received 1.2 kilobase product from we got 10 symptomatic daffodil samples, and 9 of them were producing the product, and no amplification was detected in 1 symptomatic and 10 symptomless control group. And then we moved to conventional RFLP analysis, and we use SSP1 and RSA1 because we need to differentiate it with European stone fruit disease and apple proliferation or pear decline. So our results show up, the digestion patterns are resembling phytoplasma, mali, or apple proliferation symptoms. So we moved to the virtual RFLP analysis, and for reference we use the group 10, phytoplasma, Candidatus phytoplasma mali, and we get 3 different digestion patterns. Just to mention about, we have 9 symptomatic ones, and according to them, we call them as Narcissus 1, 2, and 10, and 11, and 6, and 7, and 9. As you look at the digestion patterns, one of them, our symptomatic Narcissus plant, NAR1, NAR9, sorry, is missing MS1, MSE1 site if we compare it with reference strain. Interestingly, it is also missing the TAG1 site, and it is resembling an SSP1 additional site. And all of them are showing another HNMF1 site. So we have to give an idea about, the digestion patterns are different, but in the phylogenetically, which clade or which family they are belonging to. So we included 10, group 10, A group, and 12A group, which is also the Phytoplasma solani strains from Iran, which I explained previously, and Narcissus tazetta yellows disease, which is the peanut witch's broom. So all of our isolates were falling into the clade of group 10A. So interestingly, we were thinking about, okay, it is in the group 10, so how about the host versatility? And we look at our phylogenetic tree in a closer manner, and we see from Phytoplasma mali isolates from Turkey, Poland, and Czech Republic, which are showing the host as apple. And another one is Candidatus Phytoplasma mali from Czech Republic, and which is the host is sour cherry. And another one is from Lithuania, as the Sprea flowering. And then we have Candidatus Phytoplasma mali, the host is pomegranate. Interestingly, one of our isolate, NER6, is showing a high similarity with Candidatus Phytoplasma mali from Slovenia. The host is apricot. Okay, and then we move to the light microscopy detection, and we try to find out the localization, and we try to confirm our results with microscopic methods. First step is the vascular bundle determination by using safranin O and the alisanin blue staining, and you can see the slides of the vascular bonding of Narcissus tazetta. And by blue, you can see the phloem region, and by red, you can see xylem region. And where we need to find phytoplasmas is in the phloem tissue, of course. Then we do the DAPI staining of our slides. And then from the negative control results, we didn't detect any fluorescent intensity in the phloem tissues. And in our sample from the NER1, we get the fluorescent intensity in the phloem tissue, which is showing, this is not specific for phytoplasma, but somehow there is an intensity in the phloem tissues. And next, we move to the transmission electron microscopy to see whether our Narcissus tazetta tissues are infected with phytoplasma. And interestingly, in the area of phloem, we can see the sear tubes and the chloroplasts. And we see some inoculation in this part. So we focus on the region. And you may see some pluriformic bodies in the phloem vessel. We need to do maybe gold staining on this one, but still we can detect some of the phloem vessel showing fluoromorphic bodies. They are showing some typical wall-less cells like phytoplasmas. So as a conclusion, in our study, among tested 10 daffodil plants, we found 9 were infected with Candidatus Phytoplasma mali. Although we know group 10A subgroup phytoplasmas have been reported frequently from the woody plants, to our knowledge, this is the first report of Candidatus Phytoplasma mali infecting daffodil plants worldwide. And we confirm our study with light and electron microscopy. And we see, 10 daffodil plants and 9 were infected. It's a high infection rate, so only in a small number of tested daffodil samples indicates that the incidence of this pathogen might be very high in daffodil plantation. So we need to think about phytoplasmas are mainly spread by vegetative propagation and insect vectors. So the risk of outbreak of phytoplasma diseases in vegetative propagations, like daffodils, in the future it could be facilitated by using infected propagation materials and discovery known and possible vectors for group 10 phytoplasmas. So with that, I would like to thank my new and young team in Istanbul University and our longtime mentor and collaborator, Professor Kadriye Çağlayan, for invaluable discussion and all help. And I would like to dedicate this talk to our professor, Dr. Mona Gazel, which we lost in the terrible earthquake. Thank you for listening.
Thank you, Isil, for this nice presentation, and thank you for taking care of these beautiful flowers, that they will be still available for future Wordsworth-like poets. And I'm open the discussion.
Thank you. Thank you for your presentation. Interesting. I was interested also in reading your standard abstract because I think apart from one report from Czech Republic, if I am not wrong, Jana is here, she can correct me, apple proliferation out of woody plant is almost never reported. I have a technical question. When you show the phylogenetic tree, I could not see where was the Candidatus phytoplasma mali reported as such. So the strain type is there, or you just took all the Candidatus in general, just to have an idea of the angle of the differences, since it is an herbaceous host. It is in this?
Here it is the Candidatus phytoplasma mali, the reference 80 strain.
Okay.
Okay. It is belonging to the same clade, and if we look at other group 10, Prunorum and Pyri is here, if you can see.
From the phylogeny, the tree cannot be distinguished too much. Okay, okay. Do you have idea, because the bulb usually are imported. So a possibility for an apple proliferation in this species can be from abroad. You are thinking it is from some local infection. You have idea about if the bulb were from Turkey or imported, let's say from, I say one name, like Netherlands or Poland or so on?
These are native to Turkey, and we have the same symptoms not in Mersin area, also in near Syria border. So it looks like it is spreading somehow. Maybe this is not the source, maybe it is carried with another insect vector from the source. I'm not sure about it, but I'm not confident about it is imported.
Okay, but if it is so, it is from some apple. You have apple proliferation in Turkey. The same area where these are grown, there is also apple grown?
Yeah, we have.
Okay.
But we are not sure about which one is the first to start with. Maybe the Narcissus tazetta is the source. So we are not sure. So we are also doing insect experiments, and we need to check for the origin of apple proliferation. So yeah, it should be vice versa. Yes.
Any other questions? Or some questions from the virtual world? Thank you again.
You.
And now we are moving across the Atlantic, so we have the right guy here for this part.
Yes. Now we have two presentations from the outdoor, I mean out of this door. online. The first one is by Valeria Trivellone. Valeria, are you with us? Hello?
Hello?
Valeria? Okay.
Yes? Can you hear me?
Yes, yes, we can hear you. Okay. Then the presentation in multilocus next generation sequencing of leafhopper associated phytoplasmas highlight gaps in knowledge for some phytoplasma lineages and genetic loci. I don't know the presentation. We have to put the presentation or no? Okay. We are waiting for your presentation, then you can start.
Yeah, I know, but do I need
Ah,
to
okay.
close this?
Yeah, we have to. Okay.
Good afternoon, everyone. I'm Valeria Trivellone. I work at University of Illinois at Urbana-Champaign. Today I'm joining you virtually from the United States, and I'm going to talk to you about a new multilocus next generation sequencing approach to uncover phytoplasma diversity. Here the outline for today. I'm going to start with a little bit of an overview on the main purpose and context of our research program at University of Illinois, which is not only detection and characterization of phytoplasmas. First of all, I'm going to answer this question why searching phytoplasma in natural habitats, and then I'm going to show you the importance to search pathogens using biorepositories. Third, I will present our results and then some conclusions. First of all, why we need to search phytoplasmas in natural habitats. We learned that pathogens of every kind, infecting not just crops, but also human beings, livestock, and wildlife that human beings depend on socioeconomically, colonize a large range of habitats. I want to mention J.R. Audi. He was a medical entomologist, and in 1958, he recognized that the distribution of a pathogen is wider than the disease caused by it. This is a really fundamental point when we start to think about how diseases present themselves in places and hosts that they were not being known before across the biosphere. Crisis of emerging infection diseases stems from at least the lack of three things: comprehensive inventories, deep phylogenetic context, and last but not least, a broad landscape context, including the mosaic of habitats. We know that focusing on agro-ecosystems does not provide the whole information we need to assess the risk of emerging plant diseases. And we also know that prediction is much less expensive than reaction to the crisis, and it is a matter of national concern. We provided this scheme named after Audi, representing the operational dimensions to assess the risk of emerging infection diseases. We can use it to have a much better idea about the distribution of pathogens and diseases across the biosphere. This Audi space will look at the complex ecological arena and how interfaces are involved in pathogen distribution and circulation. As you may want to see from this picture here, we have three habitats: agrospaces, urban habitats, and wildlands. And then the interfaces among them where there is a potential for pathogen circulation. And all of this has been driven within a matrix of time, the blue buffer here, and within a climate matrix, the yellow buffer. That are influencing the structure of the interfaces. We must understand that most of the time, and in many places, including natural habitats, pathogens are silent. In fact, they are still there when the disease is not apparent. If you want to know more about the literature cited in this presentation, you may want to scan the QR code on the top of this slide. For phytoplasmas, a large literature is available focusing on the agrospaces. So based on a literature review, we found that about 10% of previously documented associations involve non-cultivated plants. Almost all known associations were documented in anthropogenic habitats and from plant hosts showing disease symptoms. Almost all known vectors were documented in agroecosystems or surrounding rural habitats, and very few attempts were made to document phytoplasma association in natural areas. Now the question would be why we need museum biorepositories and archives to study pathogens and their dynamic across the biosphere. So the answer is because information and physical specimens can be accumulated over time. We can come back to the physical specimen that we call usually voucher to reevaluate taxonomic status and reevaluate molecular analysis. These inventories may generate geographically extensive and site-intensive field-based inventories of communities. They also provide cumulative digitized information, and this basically represents the baseline for recognizing changing interfaces across the biosphere in natural, in managed and agricultural ecosystems, and the cross landscape from regional to global scales. In this picture, we summarize this concept, and you can see that archives fuel the DAMA protocol. DAMA stands for document, assess, monitor, and act. And this is a new protocol that we have been using lately to cope with emerging diseases. The biorepositories and archives are the core of this predictive protocol that we also recently applied for phytoplasmas in a regional study case in Germany. You may want to find the citation here. In 2018, we started our research program at University of Illinois using a large biorepository available at the Dietrich's lab, where over 3,000 bulk samples are preserved and were collected in natural areas. These samples were mainly used to carry out research in systematics and phylogeny of meter insects. But in the last 5 years, we start to include research on plant pathogen. For all these samples, we have detailed sampling event information. Here I mapped the coordinates of the collection into QGIS to carry out a raster analysis, which revealed that the sampling points are mainly located in natural habitats. You can see here the black pixel. In the last 2 years, we published 3 papers with the last one on the left, including a novel molecular approach to uncover phytoplasma diversity. This is the overall workflow of our research project, and today I'm going to focus on the central orange part of this workflow. We used individual insect sample from our collection, and then after DNA extraction, we carried out an initial screening using qPCR, and then phytoplasma positive samples are further analyzed using a fairly new high-throughput sequencing approach called anchored hybrid enrichment to collect data from several loci, which can be used for classification and that may inform phylogenomic studies. So briefly, this technique that we acronymize as HAE consists in the synthesis of short oligonucleotide sequences that are complementary to target regions in the genome and hybridizing them to a DNA library and isolating the targets from the genome before the high-throughput sequencing. So there are several advantages associated with this method, and in this picture here, retrieved by Lemon and Lemon 2013, The advantages in using the HAE, which is this on the top, as shown, and as you may want to see, comparing to other technique, including the whole genome approach, HAE is much more, perform much better than other. One of the advantage of HAE is that you can incorporate variation of target loci based on what was already published. Our research team developed 2 probe kits. With the first probe kit, the main purpose was to prepare probes for Auchenorhyncha insects, and as an add-on, some probes target descendants of distantly related bacteria. The probe kit allowed to resolve the relationship among distantly related Auchenorhyncha, but also we were able to detect descendants and phytoplasmas. Here I summarize results from the first probe kit. This is the maximum likelihood phylogenetic trees of phytoplasmas based on 16S gene, and in red are included over 30 phytoplasmas strains detected in these last 2 years working on our biorepository and using the first probe kit. We detected strains that are closely related to all the major clade of phytoplasmas, as you can see here, from 1 to 7. Maybe the most important results from this pilot study was that HAE generally outperformed traditional nested PCR and Sanger sequencing. Another result that is important to point out is that we recorded 3 new groups and 8 subgroups that were described and are listed in this table. And the new groups and subgroups were from all over the world: Zambia, Australia, Kyrgyzstan, China, Switzerland, United States, Thailand, Madagascar, and Taiwan. Here, the same information reported on the map. I wanted to show you that another important result is that we documented and we recorded for the first time 30 new potential insect vectors. One of them, you may want to see here, new for the science, a new species for the science. With the second probe kit, we incorporated more phytoplasma loci for a total of 129. Here I'm showing you preliminary results, and overall, HAE performed well at capturing data for 6 housekeeping genes. We prepared this phylogenetic tree, including which is for the 6 concatenated housekeeping gene, and we included 5 reference phytoplasma strains and 3 more outgroups, including Aculeplasma palme and Firmicutes. And this phylogeny is largely congruent with previous comprehensive phytoplasma phylogenies already available in the literature, and moreover, strains belonging to well-studied phytoplasma group, such as, for example, group 1 and group 5, were better supported than other phytoplasma groups, such as group 11 and group 14. And this is pretty, it's pretty clear, and it is an indication that the more data are available in the literature, the more the phylogeny will be, the results will be well supported into the phylogeny and more accurate. Overall, the phylogeny was well supported, and the classification was more accurate when we compare these results with the results obtained from the first probe kit that target mainly the 16S gene. For the future, we are planning to increase the number of samples and reference strains, and then we are comparing phylogeny with single gene, including 16S. We are including sequencing data from other more variable genes, for example, IMP or VMP, that we were able to retrieve using HAE approach. And we are also evaluating results from 4 plant loci, and preliminary data indicates that all of these samples include genes from plants that more likely have been ingested by these insect vectors, or by these insect species. To conclude, this study should enable us to evaluate more fully the variability of multiple genomic loci and clarify some silent association in natural areas that we are not aware of. So finally, we all know that plant pathogens are mostly well studied from the perspectives of plant disease epidemiology and management. We also know that plant pathogens circulate in natural habitats silently, and that gap of knowledge on phytoplasmas in natural habitats makes new emerging infection diseases unpredictable. So this is why we are proposing a novel molecular and operational approach to document the diversity of phytoplasmas, and this data can be used in a new proactive protocol, which we call the DAMA protocol. Thank you, and if you have any question, you can reach me at my email address.
Thank you, Valeria. We are waiting for some question. You can hear now, Howard, yes? You may? Valeria?
Yes?
Yes, okay. This is a question arriving now.
So, can you hear me?
Yeah, I can.
Okay. I saw the data, but there are no phytoplasma strains from India. Any particular reason?
There are no strains from India because, if I remember well, we don't have specimens from India, collection events from India in my lab. I think this is the reason. I have to double check because we have more than 3,000 bulk samples, but as far as I know, we don't have any, unfortunately, collecting event in the last 20 years from India.
Okay. So, oh, sorry. I mean, I was actually assuming that this is a data, not the specimen. So, the entire data has been done by collecting the actual samples from all the countries.
Yeah, basically, the group, the team here at the Dietrich Labs work all over the world, mainly in certain countries like China, where we have established collaborations, South America, Central America, Africa, where we have like established collaboration. So for this country all over the world, like Australia as well, we have many collecting events from several countries, but of course there are countries that my team was not able to reach, unfortunately. But this could be a good opportunity to establish some new collaboration.
This is a very huge project, very interesting. Yeah, another question?
Yeah, so another question is about group 11, which was not fitting with the conventional phylogeny. Am I right to whether I have understood it right?
Sorry, could you repeat the question?
In your presentation, you said that this particular new method that you are employing, the group 11, doesn't fit with the conventional phylogeny. Did I understand it? Did I understand it right, or what it is?
As I mentioned, this new approach is based on a probe kit, and the probe kit is based on what was already published. So when the probe kit was prepared, we used some reference strains that are available. So the reality is that this method worked pretty well with well-known groups that for which we have a lot of information already published. Performed less better for groups for which we have less information. This is what was the main message. So we have been accumulating new strain for this less studied group, like group 11. So we hope that we are uncovering much diversity that may be incorporated in new probe kits in the future.
Okay, thank you. I have a question, Valeria. In your workflow, you mentioned that you use them or will use a non-destructive DNA extraction. What this means?
Sorry, a new DNA extraction?
Yes, no destructive DNA extraction.
No destructive. Yeah, this is pretty important because, as I pointed out, something that we are routinely missing, but not just in plant pathology, I would say in general in pathology, is the importance of vouchering. So using non-destructive DNA extraction means that I can come back to the physical specimens. So we basically perform extraction of DNA on the abdomen, clearing the abdomen, and then we keep the whole specimens, we voucher the specimens, we keep the abdomen after the extraction, and we deposit the specimens in the collection. We are able to come back to the specimens. This is really important.
Okay, thank you. Okay. Thank you very much, Valeria.
Thank you.
Thank you for staying here. And goodbye, Valeria. Thank you.
Yep. I can stay here for the next presentation as well as are my two students that are not present today. So eventually, if there are questions, I'm here.
Okay. You will stay with us. Well.
Yeah.
Okay, so the next presentation from the Illinois is Preventing Phytoplasma Emerging Diseases, Phylogenetic Relatedness and Landscape Analysis to Assess the Risk of Outbreaks, from the same group, and the main author is Christine Fink.
Hello, my name is Christine Fink, and I'm an undergraduate student at the University of Illinois in the United States of America. Lisa Kwan, a fellow undergraduate, and I will be presenting our poster, Preventing Phytoplasma Emerging Diseases, Phylogenetic Relatedness and Landscape Analysis to Assess the Risk of Outbreaks. This study aims to assess the various factors that increase the risk of phytoplasma infection in croplands.
The study has the main objective to assess the factors increasing the risk of phytoplasma spillover from natural habitats to croplands. Using stratified random strategy, we selected and extracted DNA from 191 insect samples collected in natural areas. We carried out qPCR for the presence of phytoplasma. We used ANOVA test, general linear, and Bayesian regression models to determine whether geography, phylogenetic relatedness, and trophic specialization of the insect host influence phytoplasma positivity rate. Landscape analysis was carried out through QPIS, and we have two main objectives. The first one analyzes how phytoplasma infection is affected by biogeography, phylogeny, and trophic specialization of the host insects. Considering the average values of CQ, the grass specialists in Phylogenetic group 3 averages a CQ value lower than 32 and therefore more likely to be phytoplasma positive. Based on the interaction model, phylogeny of insect host interacts with biogeography to influence phytoplasma infection rate. Using a logistic regression for the presence or absence of phytoplasmas, we showed that biogeography, phylogeny, and trophic specialization of the host insects and their interactions affect the presence of phytoplasmas in the insect samples. Based on the Bayesian regression model, there's a higher probability for samples originating in the New World to be phytoplasma positive. For samples in the Palearctic Oriental region, there's a higher probability for phytoplasma positive samples, and tribes belonging to phylogenetic group 1 are more likely to be infected by phytoplasma. Those belonging to group 2 are more likely to be phytoplasma negative.
To answer the second objective, we used QGIS landscape analysis to analyze the risk of outbreak from 2 positive samples. The map to the left shows that the sample was collected in a closed forest and several scattered small croplands were present in this landscape. The sample in the map to the right was collected on a natural grassland of sparse vegetation, and no cropland within the 12-kilometer buffer was present. Therefore, the risk of outbreak is much higher in the landscape in China, which showed higher cropland edge density, higher like adjacencies, and patch cohesion indices. Overall, this experiment demonstrated that phylogenetic relatedness, geographic origin, and specialization of hosts have a significant effect on phytoplasma interaction, and that tribes from phylogenetic group 1 and grass specialists from group 3 are more susceptible to infection in Palearctic Oriental regions. We also found that landscape composition and configuration may affect the probability of phytoplasma spillovers. Overall, these results may be helpful with predicting emerging phytoplasma diseases and preserving global food security. Thank you for your attention.
Thank you, ladies. And we have some time for questions, if there are any. If there is no additional questions, thank you again to America. And I would like to thank the audience. I hope that you enjoyed this journey around the world as much as we do here at the chair table. And it's time for coffee break.
Yes, thank you. Thank you, Valeria. Bye-bye.
Thank you very much.
Bye, thank you.
Okay. Now we can go to coffee break and then we'll come back here again. After.
So a very good morning. Welcome to all IPWG delegates for today's morning session on phytoplasma disease of quarantine and relevance. We have 5 presentations in this session, and I request all the presenters to please keep well in time so we can at least 2-3 minutes for discussion. That is again very important. So first, I would like to thank the organizers to giving us this opportunity to chair this very important morning session on quarantine relevance. You know, quarantine is very, very important in the management of phytoplasma disease around the world. And I would like to request my colleague Gulnara to please start the session.
Okay. Good morning, everybody. And now we open the session Phytoplasma Disease of Quarantine Relevance, and I would like to invite first speaker, Ajay Kumar Tiwari, to present of the books Phytoplasma Diseases in Asian Countries. Please. You have 20 minutes.
Thank you, Gulnara. I must say thank you again to the IPWG for this opportunity and giving us a space to release our book at this important platform. Can we have the slide, please?
You can open.
No, I have. It's in a slide mode. Can we have the slide, please? Thank you. So this is a 3-volume book series which is published in last month in online mode by Elsevier, our known academic publisher. And this 3-volume book series was planned in 2020 when there was lockdown in all parts of the world. We were facing COVID-19 pandemic. So at that time, I was discussing with my senior and mentor, Professor G.P. Rao, so how to utilize this time. And he was telling me that there are a number of good works is going on from Asian countries, like Vietnam, Oman, Thailand, China, Turkey. So, and in literature, we don't have any good compilation for these countries. So why not we compile all this information in a book form? So I connected myself to a number of good researchers across the globe who belongs to the Asian countries. And luckily, wherever I have sent my invitation, I'm thankful to all the editors and authors who accepted my invitation, and they joined the authorship and editorships. Initially, it was a one book volume, but when we submitted our proposal to the Elsevier team and the reviewer of the book series book suggested to go for a 3-volume book series. So it started from the 1 volume, we ended up with the 3-volume book series. And this is covering the distribution, diversity, status, epidemiology, management, quarantine of phytoplasma disease, what till now has been done in the Asian countries. And the authors and editors are the well-known, they are experienced one. So this is about the book. And what about the first volume? The first volume is forwarded by the Matt Dickinson. I'm thankful to him for accepting my request. The editors of the first volume is Dr. Kadria, Professor Abdullah, Dr. Mehdi from Iran, and Professor Saman Abhasinghe from Sri Lanka. So I'm thankful to all these editors also to joining me. The first volume is comprising total 17 chapters. And the first chapter is overview of phytoplasma diseases in Asian country with a mixed authorship. The authors for the first chapter belongs to different countries of Asia. And the remaining 16 chapters are the status of phytoplasma, distribution of phytoplasma, and diversity of phytoplasma of the respective country like Thailand, China, Vietnam, Myanmar, Lebanon, Pakistan, India, Turkey, Sri Lanka. So for the official release of the book, I would request to editors of the first volume, that is Professor Kadria, Professor Abdullah, Dr. Mehdi, and so please join us the stage. And also I would request to the chair and co-chair of this session to please release this book. Can I have Dr. Mehdi, Professor Kadria, Professor Abdullah?
Thank you.
Fine, we'll take together. Yeah. Yeah. Please. Come closer, please.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you, everyone. So the second volume of this series is phytoplasma diseases of major crop, weeds, and trees, which is forwarded by Dr. VV from USDA. And again, I'm thankful to Professor Kadria, who joined us for this volume, Dr. Hort from Vietnam, Dr. Subi from Oman, Dr. Nejat from Indonesia, and Dr. Gurbi from India. The second volume is comprising total 15 chapters, which are focused on diseases in cereals, vegetables, fruits, oilseed crops, legumes, palms, ornamentals, sugarcane, medicinal plants, fodder crops, and weeds. So I would request to the editors of the volume 2nd and also Dr. VV to join us for the official release of the book. Professor Subi, and also chair and co-chair of this session.
We can take together. Yeah.
Yeah.
Yeah. Yeah. Yeah. Thank you.
Thank you.
So now the last and volume of the series, that is about characterization, management, and epidemiology of phytoplasma diseases in Asian countries, which is forwarded by Professor Asunta Batasni. And I'm thankful to Professor Kenro, who joins as editor for this volume. I think he's here, Professor Kenro. Yeah. Yes. Dr. Syed Ali Reza, Professor Yupa from Thailand, Professor Suman from India. So third volume having a total 12 chapters, which are focused on the management of insect vector, full genome sequencing, diagnosis of the phytoplasma, whatever the update has been made by the Asian researchers on phytoplasma disease in their respective country. And the most important thing that I requested to Professor Rao to join as editor of all these 3 volumes, but he refused because of he was retiring in 2 years back. So he has a number of work to do. So whenever we think about the phytoplasma work in India, so you will find the first name of as a Dr. Rao. He has not only helped to the number of students from India, in India, but he has also established and given his assistance to establishing and initiating the phytoplasma work in India. Like in Gorakhpur University, in IRI, New Delhi, in east part of the India. So, and especially he has helped the weaker section of the students who belongs to the weaker section. So we are thankful to Dr. Rao, who is currently now serving, after his retirement, he is now currently serving as the director of Institute of Agriculture and Natural Sciences in Gorakhpur University. So we are thankful to him for his outstanding contribution in the phytoplasma research in India and initiating active research of phytoplasma. So all these 3 volumes is dedicated to him. So I would request again to my friend Mr. Amit, Professor Kenro, Professor Syed to join us, and chair and co-chair Professor Asunta to please join us. Please, can we have the light please on the stage? Thank you, everyone. This is time for acknowledgement. I must thanks to all the editors, authors for their valuable support and contribution in the publication of the book. I must say thanks to Elsevier team for accepting our proposal, publishing the book in time. Thanks to Dr. Rao for proofreading, editing, and suggestion for all these 3 volumes. And a very special thanks to Professor Asunta for her careful reading of the book and correction in all the 3 volumes. I would also request to Professor Asunta to have some words about this book and her experiences when she was reading the book. Please, Professor Asunta.
Thank you. I will tell you the story of this book. When Govind was asking me about this other 3 books, I said, Govind, again, we have no time to write more books. But you know, during the pandemic, everybody, at least in Italy, was writing books. So I think the time was appropriate. I didn't know what was at the end my task. He kept me busy all the winter. This is the task, revising. And I did in a hurry, so maybe still some typos there. So don't blame too much me, I did it in a hurry. But in any case, I think this book, why is here in the quarantine session? Because this kind of information, the dissemination of this kind of information is basic for quarantine. If people is aware, they know why the problems are there, so they can take measure. So this is very important. But as it was said among us, it will be also important to translate or to use the book in order to disseminate the knowledge in the different languages of Asian countries. So everybody can read and understand, because this is really important. The farmers of today are not the farmers of yesterday. They are educated. So maybe they don't know English, but they deserve to know this important piece of information. So I think that this work that was managed very well by Dr. Tiwari will be very useful not only for Asian countries, it is also for our country and other countries, because we are feeding the world now, starting from Asia and South America in general. These are the main places. So thank you again, and I think I wish all the luck. Sell 2,000 copies. Okay, thank you.
Yeah, I would like to say, Ajay.
Thank you, Professor.
I would like to say something.
I would also request to Professor Rao to have some words.
Thank you. First, I would like to thank all the editors and potential authors to bring this very three volumes, having very updated information of all the important and emerging diseases in Asia. You know, Asia is very wide one, have a very diverse geographical location, plant diversity, crop diversity, and it's really, I'm very happy that most Asian countries now, most of the active plant pathologists, they have started working on phytoplasma diseases. And now we have a very, very strong team, especially in Turkey, Iran, India, Thailand, and Japan. There are really very potential labs are there, senior authors, senior scientists. They have been working on very, very important, and that is not limited, information is not limited to Asia. This all information will be very, very useful for other countries, because most of the diseases, they are very common in European countries, American countries, and rest part of the world. So I must thank Dr. Tiwari and other editors and all the authors for compiling very, very important information, and I think this volume will be very, very useful as a source of, for students, PhD students, scientists, and all plant pathologists and quarantine people who are involved in phytoplasma research. Thank you again. And I'm again very, very thankful that I'm really honored that these important volumes are dedicated to me. I'm really feel honored and thankful. I will feel obliged to be involved in all the activities in IPWG, related activities. Even I'm retired, but again, I've been working in university and would like to be associated in any activities for IPWG and phytoplasma research all over the world. Thank you very much.
Thank you, sir, and thank you for your attention. Thank you.
Thank you, Ajay, and I would like also to thank all of the authors, Assunta Bertaccini, Kadriya Chalayan, Ali, Govind Rao, for these useful books. And I believe that now, after this presentation, audience would like to get these useful books. And now, next presentation from Professor Assunta Bertaccini. From Assunta Bertaccini, Candidatus Phytoplasma aurantifolia and Candidatus Phytoplasma australasiae, epidemiology meets quarantine. Please.
Thank you for the time now. I hope I will be enough clear. After the dinner of last night, maybe I lost some, let's say, clearness. This presentation was done just searching in the database, starting from the awareness that there is some confusion when we are giving name to the phytoplasma, especially in the gene bank, where everybody of us is submitting sequence when we have them. Just to remind you, everybody is following the Candidatus Phytoplasma designation guideline published in 2004. But after almost 20 years, the group that is part of IPWG and part of IOM, you hear mention this word, maybe you are not familiar. IOM is the International Organization for Mycoplasmology, and it is enclosing also the plant's phytoplasma, so the plant mycoplasma, so the phytoplasma. So this group, that is several of us are here today, you can see, published 1 year ago some revision about the designation of Candidatus phytoplasma species. Why a paper on this? A paper on this because at least in the quarantine, European quarantine that is led by APPO, they use this classification, not the subgroup or group. And I think also other quarantine. They are used to have Latin names. So names is what is better perceived by other people. Therefore, in this revision, I think you can find it is not open, but in case you cannot retrieve it, ask us, one of the authors, and we have copies, so we can send it to you, because something was changed. Here is a piece of the provisional. I must remind you that the classification we are using, both of them, are still provisional classification. They are not officially accepted as taxonomical rule. Here you see a comparison between the ribosomal group and the Candidatus phytoplasma. I hope to be clear, because all my students are not understanding well when I explain this, so I try to be more clear with you. Both this provisional classification is made on the same gene, that is 16S ribosomal gene. The difference is the Candidatus phytoplasma is relying on identity percentage, so sequence, while the ribosomal group is relying on restriction fragment length polymorphism analysis. So the information are usually the same, but not always. Just from this list, I marked in red the two candidatus, of which I will explain a little bit more now. Candidatus Phytoplasma aurantifolia, the first one described here in Oman in 1995 by Monique Garnier-José Bové group, and the Candidatus Phytoplasma australasia, that is also present in the 2004 paper, and for which at the time the author, I am maybe one of the few still working of those authors, decided to leave it because even if it was the same, so at the time we were thinking subgroup and candidatus may be the same. Now we know it's not. And so we left the candidatus australasia because we were saying maybe it is important. And as you will see, it is important. This is the remaining list made in the same way, the Candidatus Phytoplasma and the ribosomal group. In the middle is the name of the main disease for which this phytoplasma was detected and identified. On the top of this, there are 3 candidatus published in Australia, one in Papua New Guinea, for which there is no correspondence with any ribosomal group. But doesn't matter. Ribosomal group are giving us information, but I think in the future we should more and more try to rely on Candidatus Phytoplasma designation. Just following the updated rule that you see here. Now we changed the threshold. Before it was 97.5, now it is 98.65. This is not from our imagination, it is the threshold used in general for bacteriology. And then the other rule that is maybe not very clear here is instead of using at least 1200 nucleotide, we ask to use the whole 16S gene. Now it's not so difficult. So this is for Candidatus Phytoplasma aurantifolia, mainly slime witch's broom, and it is a subgroup, historically the one described is ribosoma subgroup 2B. While for the Candidatus australasia, it was described a few years later in New Zealand for papaya dieback. As you can see here is 16SR2D group. I listed here the paper in this meeting that are dealing with this phytoplasma, at least checking from what is written in the standard abstract. I will not read now, we have no time, but you see we have at least 5, but probably there are more, all dealing with this 2D, I mean, Candidatus Phytoplasma australasia. So lime witch's broom is still very important as Candidatus aurantifolia, but apparently Candidatus Phytoplasma australasia is climbing. There's an old paper published in Australia writing a successful phytoplasma in Australia for this one, australasia. And I think this is true because we are starting to detect it until Egypt and scattered already also in Italy, I don't know, in other parts of the world. And I don't know on the opposite part of the world. It's quite spreading. So I like to be short. I just was searching because Professor Rao was telling me when we have to call aurantifolia, when it is australasia, but this several years ago. I searched in the gene bank, and I searched using the Candidatus as a comparison, and just using the general sequence, I was getting something like 4,000 sequences, just for these 2 Candidatuses. Then I restricted to the taxid, I mean, just giving the Candidatus aurantifolia and australasia name, and the number, as you can see here, were reduced. Then I verified the sequence for the identity threshold. It is a play, but you see that above 98.65, we have for aurantifolia a number of appropriate sequence, but there are about almost 300 that are below threshold, or in the, yes, also below the old threshold. For Australasia, the situation is not so crowded, but still we have something that is not fitting the rule, generally speaking. You can read all the numbers in the abstract book. I will not repeat now. So I did more. This is still more confused, but you have it in the book. I took, because there is a lot of sequences, I took 5 from different situations, let's say, identity, and I did this phylogenetic tree. What I was able to verify, I don't know if, yes, you can see, I think, the color. You see the Candidatus aurantifolia is down and the Candidatus australasia is up. But you can just see that there are some aurantifolia down and some australasia up. So there is some wrong naming in the gene bank. And also, looking at the last column with the identity in blue is to australasia, the identity in green is to aurantifolia. And if you look carefully, look in the book, we will not go through it now, but generally speaking, there are cases, and you see here marked in yellow, in which also the threshold is not telling you which is the correct naming. In several cases, they are both above the threshold, and so you don't know if it is one or the other. So in these cases, you can say, okay, we go to the subgroup. This is the best. But you can see here, the subgroup is not always fitting with the candidatus. Also for subgroup, there is some, let's say, not a very clear situation. The number you see in parentheses is that, for example, 2D is not exactly 2D, is 0.97 identity. We have Wei here, she did all these rules for this. So, informatic rules. This is just something to make you aware of this problem, but I don't want to leave you with the problem. So, following the paper I showed you, you can see that we are suggesting when you have trouble in detecting or determining the Candidatus phytoplasma species, to add other gene, especially housekeeping gene. So here you see for the aurantifolia and the australasia, the one we are suggesting, and you see the gene bank numbers are available. So the important thing is the comparison with the aurantifolia described initially. Otherwise we get lost. Also, when you write paper, use the strain that is the Candidatus originally described as comparison. Otherwise, everything can be not clear or not correct. If you look down to the australasia, you see another situation. If you read carefully the paper, you can see all this in the paper. In any case, the Candidatus australasia originally described is only available as 16S ribosomal gene. We have this situation also for other Candidatus phytoplasma. And what the group did for this was to select an alternative strain for which we have more gene available and that was 100% identical to the one described on the 16S. We have no other tool than 16S in this moment to do this differentiation. So you will see for our aurantifolia, we do not have the 5 gene listed, we have 3 only, but this is already something. You will see that this is not enough, but this is tomorrow's presentation. So, this is the first step. But my, let's say, final, or not final, but my suggestion in this moment of the research is, okay, we should follow this guideline. We can also, as some people lucky with money can do the whole genome and the any differences, all these kind of things that are not very handy for everybody. In the future, probably it will be a click also for this, like is PCR. I witnessed at the start of PCR. It was like the start. Now it's nothing. So probably also the sequencing will be like this. And probably the classification or the differentiation will follow. But really it doesn't matter. If we use 16S, if you use other gene, if you use the whole genome, we only know genomic data. We don't know phenotypic data. So we are still candidatos. So really, what is the aim of this? Also for quarantine purposes, because quarantine will never rely only on the 16S or genome. If they do this, it will be very difficult. As a plant pathologist, we know that the main issue is pathogenicity. And instead, we have some idea for some phytoplasma, but this is not clear. There could be a faster solution if people start to work on this. We, in Bologna, since 10 years, but I started 1983, not 10 years ago, together with Dr. Contaldo, who is here, we were able to isolate and produce colonies containing phytoplasma in artificial media. What I show you is the second paper, because the first paper we published was not having the recipe to do this. This is having all the media we used and we are using. So everybody can try just following this 2016 paper. And this is still in that paper. You see these are how they look, because these are pictures of the colony under the binocular microscope. And these are the dishes. And these are already too much grown. When they are very, very small, you will see more tomorrow about this, it is possible to detect, even if it is complicated to detect here. But the same problem is faced by mycoplasma. These are always bacteria difficult to culture because they live in isotonic environment. But what we need is to merge with other competence, with bacteriology, with other scientists that can help us, especially in improving the media for growing them. Just last, I want to mention you how many people since 1989 worked with me, but it's not like this. I put in yellow the person who are still on phytoplasma. Yesterday, my student from India presented the seeds that are transmitting phytoplasma. These are the seeds that are working, I hope, on phytoplasma. They are all younger than me, so I think they will continue. And in green, the piece that is most important in all your laboratory at home, my technician. She is with me from 1989, and I will retire before her, but I will keep her in job in some way. Thank you for your attention. I hope you understood it was not, okay.
Thank you, Professor Bertaccini, for this interesting presentation. And are any questions from audience? No, maybe from online participant? No. Thank you. Thank you again.
Too difficult for question.
And now, next speaker, Professor Nicola Fiore, with the presentation, Multilocus RFLP characterization of Candidatus phytoplasma piri strains in pear from Chile and Italy.
Okay, good morning everybody, and thank you for the host of the meeting, the Sultan Qaboos University, and the organizer, Ali especially. And I will talk about a work that we are carried out, as I'm carrying on, in collaboration with the University of Bologna, Alma Mater Studiorum, and the University of Chile, in which I'm working now. But the University of Bologna was my Alma Mater Studiorum too, because I studied my undergrad there. And especially the collaboration with Assunta, and this is a part of the work developed by a student from the University of Bologna that we made a stage in my university for develop his undergraduate thesis, Pietro Bianco. The Per decline was found in Europe, in Italy, about in the year 60, and detected in Chile in 2017, or reported, was detected before. And the idea of this work is see if there are some difference between the strain found in Italy and those found in Chile. The symptoms, just a few pictures, but you know them. In Chile, you can see a plant with reddish leaves during the period at the end of summer or during all the fall season. And normally, the plant with the reddish leaves is isolate or a small group of plant. In the case of outbreak, we may see more of this plant with symptoms, of course, but normally you can see one plant there, another here, with this kind of symptoms. The most important that you can see in the field. Of course, this reddish leaves may be appear for other problems that may be correlated to other abiotic or other biotic or abiotic stress in the plant. But it's a good indicator to take samples from this plant and analyze it in the lab. We in Chile found some necrosis in the vessel, epinasty in the red leaves, or you can see different intensity or red in the leaves. And of course, the production, the yield is very, very low with a low quantity of fruits. Well, we take 20 samples, 10 from Italy and 10 from Chile, from symptomatic plants, and then carried on the right PCR with the 16SR gene and nested PCR in the same gene, and then FLP, I can use the FLP in vitro, no, with this restriction enzyme. The positive samples were analyzed for different other genes, the most important, the most used in this case, or the most present in gene bank too. And the nested PCR, by the nested PCR, we realized FLP with the other 4 genes. Finally, something that I had that yesterday, because I received the last result just during the dinner, the sequencing and MLSA analysis with some of the amplicons obtained with several genes. Okay, the results in the 16S ribosomal gene, we obtained 10 samples positive out the 20 analyzed. This is normal with phytoplasma. We know that despite using a lot of repetition of the sample, we may obtain false negative, sometimes false positive, but in the case 7 was positive from Italy and 3 from Chile, and we performs the RFLP with these two enzyme, restriction enzyme, and confirm the presence of the 6ESR10C. Then, using the other gene, in the next PCR, we found 7 out of 10 samples confirmed positive with these genes, 4 from Italy and 3 from Chile. Now, we may amplify these samples, these in the marker, PN10, and completely with all the gene, okay, from Chile, and we used Candidato Phytoplasma mali half-out group to as a control. Then only PN13 we can amplify 5 genes out of 7, and finally with PN11 and PN14 from Italy we obtain 3 amplicon out of 6 used in this case. We now focusing in the restriction enzyme. We obtain the same pattern at the same letter, the same pattern of the profile, of the restriction profile, and you can see in this in Liu ASF as the Candidato Phytoplasma mali present a profile different from all the samples from Candidato Phytoplasma piri. In this case, SEC-A and ESU-3CP, we have a problem because Candidato Phytoplasma mali, in this case, I am afraid to change the slide when I show this, but we have the Candidato Phytoplasma mali that present the same pattern that the samples from Chile, and in this case from 1 from Chile. The better profile are obtained where with SEC-Y and TAF, but unfortunately we do not obtain amplification for the sample PN14 from Italy. Finally, we use for multi-locus sequence analysis only the 3 gene ASF, SEC-Y, and SEC-A that were completely totally amplified, no, with the unique exception of PN11 that only we obtain 3 amplicon out 6. And then the MLSA was made with these 3 genes and all the samples not included PN11. Well, as you can see now in this phylogenetic tree, concatenated these 4 genes, including 6ESR-RNA, we obtain a separation between the strain from Italy and the strain proceeding from Chile. And of course, the Candidato Phytoplasma mali is separated from the strain of Candidato Phytoplasma piri. In conclusion, the analysis with MLSA confirms that was observed with the multilocus RFLP are the same. In this condition, with this number of samples, of course, seems to be a geographic differentiation between periodic line phytoplasma isolates. And of course, it is important to expand the work using a lot of number of strain proceeding from Italy and Chile. But the indications are very clear. Maybe the geographic differentiation would be associated to a different environmental condition. Chile is not the same as Italy. And of course, the different vectors. Because I remember that we in Chile don't have
Cacopsylla piri, don't have Cacopsylla piricola, and neither pirisuga. But we have Cacopsylla bidens, and in the epidemiological study that we are carrying on now, we found a lot of other psyllid that have to classify. I think that this psyllid that I found now are not classified yet. This is the great problem. And of course, with this new psyllid, we are carry on. We started the transmission trials to confirm if they are able or not to transmit the phytoplasma associated to Candida phytoplasma piri. One of the goals of this work was to have another technique that we be able to characterize peer-disease phytoplasma strains without sequencing before. And then this is easy to finish the work in the lab without send the sequence, the amplicon to an agency that can sequence these amplicons. But it's very interesting. Note as there is a geographic differentiation, maybe due to which I told before, and it's very important to complete this work with other strain. Thank you very much. This is a work supported by a project financed by the government of Chile, the government of Chile, about epidemiology of peer decline in Chile. It is spreading. There is not really outbreak now, but the plant with the problem are growing in the field in different part of the country in which pear is cultivated. Thank you very much.
Okay, now this talk is open for question. Luckily we have time for 3 questions. Yeah, Dr. Kadri first.
Thank you very much for your nice presentation. I was wondering if there was any difference on the symptoms between Italy and Chile. You know, the isolates look like different. I was wondering about the culture and also about the symptoms.
No, there is no difference between the symptoms in Chile or in Italy, and we have more or less the same variety. There are cultivars.
Do you have any quick decline? Quick decline due to peer decline?
Decline?
Decline,
yes.
Quick decline.
Peer?
Peer, yeah, declining of the
peer
trees.
Decline. Yes, of course.
No, no. I mean, there are some strains which cause mild symptoms and some of them are severe.
No, yes, this is a news because we are writing about this, but we found other phytoplasma, we speak of them tomorrow, that infected pear in Chile that cause the same symptoms of those by 6-10C. Is other pathogens you ask for it, they are not associated to peer decline in Chile, or symptoms similar of peer decline.
One other comments, maybe, you know, we were also studying.
I can.
Okay. We are also studying the vectors in our country for peer decline. And we detected PD pathogen also in Cacopsylla picta and Melanoneura in a very high percentage. So I would suggest if you have these ones, you know, although they are efficient vectors for Candidatus phytoplasma mali, they can harbor also piri.
Yes, no, Phytoplasma mali is not present in Chile, but no, picta neither. And then we don't know what happened, but the disease moved in the field, and we have to detect now why. We are collecting a lot of species of different psyllid, as vector of PD or Candida phytoplasma piri, but we are now making the transmission trial. We didn't found some insect, some individuals positive to the phytoplasma.
Okay, thank you.
This is important, the results of transmission trials now.
Of course, yeah.
But the vectors are surely not the same no in Europe, only in North America.
Please, Michael.
Yes.
You have.
You found a difference among your few samples, I must say, among Italy and Chile strains. But for some of your marker genes, which you showed, ACF and SECY, there are more sequences in the database. Did you check these for these differences? Whether you found your Chile sequence data, whether it's similar to European ones or not?
No, there are some difference in the.
Yeah, but in the database,
yes,
in the others.
Yes, yes, in the database there are difference. Not only the enzymatic profile is different, but the sequence too. Then when I show the phylogenetic tree, there are clear difference in these concatenated genes that we use for the bioinformatic.
Yeah, but now the question is whether you take one gene, the marker gene, as single sequence,
yes, of course,
compared to the other
yes,
sequences in the database. If you find the same sequence again, because this happened in these other multi-locus sequence studies, that you find one marker in every region.
Yes, this is true. We now are try to obtain the draft sequence of several, the draft complete sequence, the draft sequence of the genome of several strains from Chile. And this is, we may understand, comparing this with gene bank, if there are some difference. Of course.
Okay, any other queries from any online audience? Any online attending delegates? No, Nicole, I have a simple query.
Okay.
You use different multilocus genes. Did you find any, you use different multilocus genes for identification? Different multilocus genes? Yeah, so whether they are equally efficient, or did you find some very significant out of those different? Yeah. Yeah. Yeah.
No, see, a very significant difference in this case, of course.
So any particular one did you find very is very good in amplifying in most of the plant sample the phytoplasma out of different genes used? No, did you find any specific? I noticed one true 1L. I found all positive.
Yeah.
So yeah, so this is good, no?
Yes,
in
good.
comparing
of course,
to others.
Of course, of course. Thank you. Yes. Yeah. Now I would like to invite the next speaker, Dr. J.D. Sedlak, and he will be speaking on screening of European stone fruit yellows, apple proliferation, and pear decline diseases in fruit nurseries with focus on latent infection.
So thank you very much for introducing me. I would like to describe the results of our work aimed at screening of these 3 phytoplasma pathogens in Central European conditions in our country, in the Czech Republic, and the focus was at fruit nurseries and especially latent infections. So in the beginning, this is the building of our institute. The English translation of this institute is Research and Breeding Institute of Pomology, and we are a private institute. It means that we are registered in the business register, and we can act as small and medium enterprise, but at the same time we are also a research organization recognized by European Commission, so it means that we can act as in these both roles. So let's go back to the topic. It was a joint work of our institute, which is more at applied research, but our collaborators were from the Palacký University, from Faculty of Science, from Palacký University situated in Olomouc, and they have taken a more important role in the basic research, in the part of basic research. I guess you know that probably you are familiar with these two pathogens in Central European conditions. In temperate climate, they are very important. And of course, we can dare say that the competitiveness of the whole fruit growing sector depends on the ability of certification system to limit the spread and the harmfulness of phytoplasmas in fruit growing systems and in nurseries. So logically, we concentrated in our work in the very beginning or to the very beginning of the production chain. It means that we carry out our monitoring in nurseries and in mother stock of pomes and stone fruit, which is frequently grown in the Czech Republic. These were all tree species, not shrub species. And here is the representation of our collecting mission. So you can see that we visited the whole area of the Czech Republic. So it means that I guess that the representation is plus minus 75% of nursery production of fruit tree species in the Czech Republic. And there is also a number of samples which were taken in each area, and number of locations which were visited. The screening was carried out at the end of vegetation in September and October 2022. And in total, we taken 640 samples from apple, pear, from apricots, from plum, and from peach for phytoplasma diagnostics. And this is the example of the field work in nurseries. You can see my collaborator, my young, beautiful collaborator, enthusiastic for sampling and research work. And yeah, sorry, not to forget, there are also some symptoms of phytoplasma here, but it's not maybe so important. It was a joke, so you can see it here. But this photo was taken several years ago, and you will see later that the phytosanitary status of nurseries improved very much. This is the representation, the proportion of samples taken. The blue color is apple. The reason is for this such a big amount of apple samples is that concerning the Czech fruit growing, apple represents 50% of acreage of intensive plantations in the Czech Republic. And when you take into account the commercial turnover, it represents even 75% of commercial turnover in the Czech Republic. So it means that apple is very important, and the representations of samples taken by our team also reflects this fact. There were also, of course, plum, apricot, and peach and pear species or pear samples taken during our screening. this is the methodology or the methods of phytoplasma detection, and we used real-time PCR and nested PCR for phytoplasma detection, and RFLP analysis for distinction of individual phytoplasma species. And this is the core of our work. It means the results of our screening based on PCR testing, and you can see that there wasn't any phytoplasma apple proliferation detected in apple. The same was for pear decline. There was also low occurrence of phytoplasmas in plum, low occurrence in peach, but we detected 7 positive samples of European stone fruit yellow infected apricots. So, but in the general, when you calculate the final result, it was only 1% of infected trees in nurseries. Concerning the Czech situation, all these phytoplasmas are not classified as quarantine, but only as regulated pathogens. So it means that it's said by the Czech phytosanitary inspection organs that they can tolerate the infection rate up to 3%. So it means that we get very good below this threshold, and I consider this result as a very good picture of the Czech nursery production of fruit trees of palm and stone fruit species. So this is something like the conclusion or conclusive remarks. These results show that, as I said, the phytosanitary status in Czech nurseries is relatively good, but, however, it's necessary to pay constant attention to phytoplasmas in this propagation material, because especially in the case of latent infections, there is always risk that these phytoplasmas can be introduced into this certification propagation system. And of course, we must pay attention especially to latent infections, and European stone fruit yellow is example of this frequent latent asymptomatic infection. Because, so to be honest, in my career or in my work, I frequently noticed that you can have very vigorous, very highly fruiting, very prosperous trees of apricots, which are symptomless, but when you detect phytoplasma status, you can frequently discover this European stone fruit yellow infection in these trees. So it means that the logic next step would be to pay also attention to something like the tolerance in these key fruit tree species, because due to the change of global climate, we can expect that there be new stream or new infection sources, especially due to vectors of phytoplasma, which can endanger plantations or nursery propagation material or nursery stock in future. So it means that maybe you are also familiar with the field of virology. Virologists, they recognize several types of reaction. It means host versus the pathogen. There is susceptibility, there is tolerance, there is resistance, there is immunity, there is also the reaction which is called hypersensitive reaction, which is sometimes called the only functional system of resistance. So we can discuss about these terms, but I feel that we need to pay attention in future to try to establish these different types of tolerance or resistance, even if there is the phytoplasma infection in the tree, because the status or the status of the tree from the phytopathology point of view can be sometimes completely different from the grower's view, because the growers need to produce apples, the growers need to produce apricots, and so on and so on. And we can merge these two approaches together to make some economically positive result, especially for growers, because we are not scientists for ourselves, but our science should serve the public, should serve the growers, should serve the nurserymen. So thank you for your attention.
Thank you, Dr. Sellak, for very, very important talk, very interesting one. Now it is open for question. Yeah, Dr. Asunta.
Thank you for your presentation. I have two basic questions, mainly. I don't know if you can hear me. I can reach you.
Yeah, I can hear you.
The first question is the time in which you sampled, because for stone fruit, there is no importance. They are all positive all over the year. But for the other, if you sample in the wrong period, you get negative. And the second question, you mentioned nested PCR and qPCR. I would like to know how you manage, if you do everything by qPCR and then negative, then by nested, or what?
So the first part of your question, so concerning the global warming, and if you take into account that in nurseries there is very juvenile material which is very vigorously growing, so it means that in the situation of very pronounced global warming in Central Europe, the September and the beginning of October is the period of very intensive vegetative growth. So it means that we focused, we delayed our inspection to intentionally for this time. And the second part of your question, the reason for two methods of PCR was that we were two workplaces, and we tried to be economic also in our research. And our research team from Holovouci, which is situated in eastern Bohemia or in the Bohemian part of the Czech Republic, take care, or we carried out inspections in Czech nurseries. And the Moravian team from Palacký University, they inspected nurseries in eastern part of the Czech Republic in Moravia. So it means that qPCR is used for detection. This system is developed in our institute, and they at this Palacký University they used nested PCR, but both teams are world known for the reliability of their results, and in fact we are accredited laboratory. So for the Europe, so it means that I would rely on these results. Of course, not always is 100% success, but in the case of PCR, I think that there is higher probability of negative positive results than the probability that you miss some negative sample.
Thank you for the answer, but you should know that qPCR is not providing you identification. So I imagine that when you have positive in qPCR, after you run nested PCR, otherwise you don't know what is the phytoplasma you are detecting. There are very few specific There are for apple, but they are separate PCR. There is for SV, there is no for Berdecline, for example. But I don't want to keep too much time.
Yeah, but
this is technical detail.
Yeah, but we always use the method proper for the particular species.
Okay, one more question from Wolfgang Jaraunsch.
Yeah.
Thank you, because I have to make some comments on your talk and on Asunta's replies, because I've also quite a lot of experience in testing nursery mother stock material. So your period is perfect for these diseases, this is agree, but the sensitivity of testing relies on the sampling. In your small plants, maybe it's not the big problem, but this is a crucial point for the others in fruit trees. You have irregular distribution of the phytoplasma in the tree, and even in these latent infected without symptoms. So you have to have a very good sampling protocol. This is one remark. The other remark is on your last statement about the tolerant plant acceptance with a sort of amount of percent infected. You have to distinguish between plant production and the growing situation of the growers. There you can tolerate some infection, but not in the planting material. As we work on resistant rootstocks, we have this problem now in the nursery or mother stock production. If you use a resistant rootstock which confer really a resistance like an apple proliferation, then you can accept it. If you don't have it, you have always a low percentage of infected plant material spread over the region, and then you reach an orchard where it can be put up by the vector, and then you have the epidemic again. And for the nursery, this is my question now, the most important is insect vector control. How is insect vector control achieved? Because 7% what you found in SV is a lot.
Concerning the insect control, because it was a big project, and the other part of the project was also screening of vectors. So it means, but these results are not presented here because I had only 15 minutes. But at the same time, our team of entomologists also monitored the presence of vectors in nurseries. To say it in general in nurseries, the incidence of vectors of psilas is relatively or is lower in comparison with production plantations with adult trees, because nurserymen usually tends to use more strong plant protection plants, more sprays against these vectors, because they don't need to take care of residues. So it means that what my colleagues said, the vectors were less frequent in nurseries. Yeah, maybe this is the answer to your question. Yeah, but beside this, in this project, it was also a part of molecular genetics included, and our team from the laboratory complement at the same time. They try to develop the molecular detection system for discrimination of particular species of vectors. So it means that we did the work on multiple fronts.
Okay, Dr. Sedlak, thank you very much.
Okay, thank you.
Okay, and we move on next presentation. Multigen characterization of phytoplasma enclosed in 9 group infecting different host plant species. Alessandra Schivolone. I'm so sorry for
pronunciation. Okay.
Yes, Schivolone.
This is the last presentation. by our young colleague from Sultan Lab.
Thank you to introduce me. Good morning, everybody. I will speak about phytoplasma of group 9. Phytoplasma of group 9, PGMP, which is broom, are associated with disease affecting crop and wild plant in different geographic area worldwide. In the map show we have a distribution of candidatus of phytoplasma of group 9. The most common phytoplasma of group 9 are phytoplasma group 9A and 9C. Moreover, phytoplasma group 9C are associated with sesame phyllodes in Turkey and almond brooming and Kaunzia arvensis, which is broom in Iran. Also, the 9C subgroup are associated in other continents in the large range of plant cultivated and wild host plant. The Candidatus phytoplasma phoenicium is the only species described in this group, and in the Middle East, Candidatus phytoplasma phoenicium classify group 9b, and its genetic variant D, F, and G are associated with a lethal devastating disease on almond tree and nectarine tree in Iran and Lebanon. So for this work, we focus just on phytoplasma group 9 that we have in our collection, and we wanted to molecularly characterize just the subgroup 9C. So the sample of Taraxacum officinale and Cataranthus roseus collected from Italy with the symptom of virescence and witch's broom and the symptom of Leucanthemum vulgare collected in Italy with the stunting witch's broom and virescence, and sample of periwinkle from Colombia with the same symptom were analyzed to verify the presence of 9 phytoplasma and for characterize them molecularly them. DNA was extracted from a symptomatic plant collected by CTEB method, and then PCR was performed on DNA extracted and on 16SR gene and on RP, SEC-A, SEC-Y, isoleucine, IMP, and TAF gene. The same enzyme, 2R-TRU1 first, was used for RFLP analysis on the amplicon obtained for all gene, and the RFLP selected gene amplicon for differential profile were directly sequenced in both sense using the primer of amplification, and the obtained sequence were assembled, aligned, and compared to the reference strain. In 2022, the revision with line for describing Candidatus phytoplasma species were published, and the strain sharing rate 98.65% to sequence identity on 16S gene when compared with the reference strain are considered member of the respective Candidatus phytoplasma species. For the strain that showing low identity, the reference strain, is it possible to use other 2 housekeeping gene for classification? So for the PCR assays with the primer amplifying on 16S ribosomal gene of phytoplasma produces the expected size amplicon for all symptomatic sample and control strain. RFLP showed, analysis showed pattern identical to each others and to the strain in subgroup G. And the RFLP of control with the pigeon P, which is broom, taken from Lee et al., has a different restriction size. And the CK gene was not amplified in all sample, and only Naxos had a good restriction profile. The RP gene, already used for years in literature for the classification of subgroup, was amplified in all samples tested, and the restriction profile obtained are all the same, with the exception of periwinkle from Colombia. The data confirm that all samples belong to subgroup C. From the virtual, it's clear that difference between the two subgroups. Indeed, the subgroup 9C has more restriction profile, more restriction site, sorry. So these highlight the difference between the phytoplasma. RFLP analysis showed that phytoplasma strain 9C used in this work not always had the identical pattern to each other. Indeed, the sample CO, underlined in orange, periwinkle, was different always from all the other samples, probably because CO came from Colombia. Not always, but also in Naxos, underlined in yellow, the different, there is a difference with other sample, and Naxos came from Sicily. So maybe the difference provenance can contribute to this variability. Furthermore, the IMP gene of all samples was not amplified with the specific primer of Candidatus phytoplasma phoenicium, and the results highlight the genetic difference between the strain. The difference is obviously was confirmed in sequence. In fact, in the second column, all the sequence show an identity of less than 19% to the reference ones. All the other genes shown in the table also gave a low identity sequence with the reference ones. As for the 16S gene sequence of tested sample, they gave a high identity with the reference one. So maybe in this case, the 16S gene is not very informative compared with the other housekeeping gene. There is no doubt that Candidatus phytoplasma phoenicium must maintain its status, both reason for pathogenicity and classification. The most characteristic symptom associated with almond witch's broom on the almond tree are short proliferation on the main trunk and general decline on the tree with the final dieback. In the case of nectarine peaches trees, the first symptom is early flowered and flower deformation. The EPPO map of a spread of Candidatus phytoplasma phoenicium published on the EPPO website states the presence of Candidatus phytoplasma phoenicium also in Italy. Indeed, in 2019, a case was reported in Apulia in Almond Grove. The case will seem to be only sporadic. Indeed, in Italian territory, the phytoplasma of group 9C is mostly present. Almond witch's broom are associated with the Candidatus phytoplasma phoenicium and was brought to the attention the EPPO as early as 2001, and in the same year was added in the alert list and deleted in the alert list in 2006. Since the disease has spread in Lebanon, Verdin et al. in 2003 found a new Candidatus phytoplasma species, Candidatus phytoplasma phoenicium, that was found to be associated with the disease in Lebanon and Iran. Based on the PRA, pest risk analysis, was added, Candidatus phytoplasma phoenicium was added in A1 list of pets recommended for quarantine in 2017. Currently, however, Candidatus phytoplasma phoenicium is not included among priority pests. So, the sample tested in this work do not belong within Candidatus phytoplasma phoenicium distribution area. Indeed, they came from Colombia and Italy. The reporting I told before is the only present in the literature. And the difference between the subgroup of group 9 can also clearly seen from the phylogenetic tree of Hammond et al. 2017, in which it's evident that C, A, and B, and its genetic variant clustered differently. Also, in this case, the different subgroup in the tree had a different geographic origin. So the identity percentage on 16S ribosomal gene did not allow a differentiation from Candidatus phytoplasma phoenicium according with the proposed identity threshold. The identity percentage on RP gene and the collective restriction profile on the other gene tested allow differentiation, and differentiating phytoplasma is important for quarantine, and considering molecular variability, geographic distribution, and range, a different range of host species, a new Candidatus phytoplasma species could be proposed for this group. So I would like to thank my supervisor, Professor Assunta Bertaccini, our team, and Dr. Nicoletta Contaldo, and thank you for your attention.
Thank you for your presentation, and we are a little bit late, so I think we can discuss during coffee break. Are there some questions from online? No?
Okay, so on behalf of our organizing committee, I would like to thank all the presenters for a very nice presentation in the morning session.
Yes.
So now we would like,
Close?
Yeah. Now we would like to close this session, and I request Dr. Al-Sobi to announce for any information.
Thank you, Doctor.